Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. Come home to history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home.
So thank you for coming in early. So we have this next session by Dr. Luna Samanta. She'll be talking, she's a professor, Department of Geology, Ravensa University. She'll be talking on the topic comparative proteomics reveals.
Hello. Hello. So I'm just calling the names of those who have given their abstracts for young scientist presentation. Rupalin Jenna is there. Sit down, please. And Rajashree Datta. She's giving online. Yeah. Ekta Bhattacharya is there. Then Srimayi Mitra also there. And Madhusmita Datta. Prachitra Raut. Okay. So time allotted to you is 15 minutes. It will be 10 minutes presentation and 5 minutes discussion. So I will give you after 9 minutes, you adjust the clock to 9 minutes. Clock to 9 minutes. In presentation clock there. You cannot change. Or say to have no, but okay, take it. So after 9 minutes, you will have a warning. You wind up at 10 minutes, and thereafter there will be 5 minutes discussion. Everybody is encouraged to ask the questions. I would request those who are in the backbench to come to the front. I think there are enough seats in the front. So now I request Rupalin to make the presentation. Hello.
So, very good afternoon to all. My respected Professor Pradipta sir and Dr. Rukmani ma'am. So, today I am going to present my topic endophytic fungus isolated from wild rice species promotes growth promotion of the rice crops and showing defense response towards rice diseases such as seed blight. And I am working under Dr. Arup Kumar Mukherjee at NRI. So, as we all know, endophytes are the microbes which reside inside the plant tissue without causing any harm to the host plant. So, the wild rice species, especially, they have the capacity and they have the capacity and they have the major source regarding stress tolerance capacity towards biotic and different abiotic stresses. So, specially for this purpose, we have chosen the wild rice species. So, in wild rice, we have chosen Oryza nivara, Oryza rufifogon, Oryza longistaminata, Oryza birthi for the isolation and molecular characterization. So, these are the objectives: isolation and molecular characterization of endophytic fungus from different plant parts of the wild rice species, and study of the antagonistic activities towards different rice diseases both in vivo and in vitro condition, and study of the growth promotion of the rice crops and regarding the better health management of rice crops. So after isolation and molecular characterization, we found these species and all are collected from NRA only. So from different plant parts, we have found Alternaria alternata, Cladosporium oryzae, Clonostychus epicloe, Dendrophilia species, and Hypocreals, Flaviospis musei, and Corvularia lunata. So different molecular characterization like ITS and TEF and RPB2 we have did, and these all are the amplifying region, and then we did dual culture assay for antagonistic activities towards different rice diseases. The pathogen we have taken Rhizoctonia solani, Sclerosium oryzae, Sclerosium rhopsi, and Sclerosium delphini. So in dual culture assay, we have found the best pathogen against the organism or the endophytic fungus, that is EF2 and EF4, that is Dendrophilia species, and another one Cladosporium oryzae. Then some enzymatic activities, that is quantitative assay of endophytic fungus we have did, and endophytic fungus species application and seedling figure index also we have done. And we have taken the rice variety Annapurna and Satabdi. And next, work is progression on Tapashwini. And we have also did chlorophyll content and growth promotion, that is dry root weight, dry shoot weight, fresh root weight, fresh shoot weight, and root length, shoot length, and number of tillers per hill, and yield per hill also we have seen. And specially we have did expression of defense enzyme related to stress in the rice varieties. Those who have treated with endophytic fungus. So in all cases, we have seen the best organism or the best endophytic fungus we have found, that is one Cladosporium oryzae and Dendrophylla species. Then in net house condition and field condition we have trial. So we artificially inoculation the Rhizoctonia solani diseases, that is seed blight. So we have found, you can see, this is the control plant and these two are treated plant. This is EHAP2, that is Cladosporium oryzae, and this one the EHAP4, that is Dendrophylla species. So the work is progress now, and we have found, till now we have found the who is the endophytic fungus treated with that rice seed which are treated with endophytic fungus promotes the germination, seedling finger index, and growth of the plant. And apart from the defense response, we have found this the stress tolerance capacity of the endophytic fungus. And our recent investigation tries to fill the gap by isolating and identifying the endophytic fungal strains from wild rice varieties for rice health management. So lastly, the two varieties or the two endophytic fungus we have found, that is mostly we want to do further work to taking these two species, Cladosporium oryzae and Dendrophylla species. And I want to thank, or our team want to thank Professor Prashan Kumar Mukherjee from BARC and Toto Nadak sir from ICR NRI and Director of the NRI Cuttack and Department of Botany Utkal University and DST government, which is providing me Inspire Fellowship. And lastly, I would thank to my guide, Dr. Arup Kumar Mukherjee, and specially I want to thank Mr. Hare Krishna Swami, who is like my another guide and who is present here. Thank you. Sir? Sir, it's not going. Sir, one. Sir, sorry, sir. Sorry, sir. Sir, from wild rice, we have seen the stress tolerance capacity. But the current variety are full of chemical pesticides we are applying. That's why I choose the wild rice. Yes, sir. Yes, sir. But we didn't found any specific endophytic fungus. No, sir. We have found, but there is no specific character. Means we that work we have did that like enzymatic activities or different plant growth promotion capacity, but there is no significant capacity. That's why we choose the wild rice species. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Leaf of Oryza langustaminata. Yes, sir. We have tried. No, sir. No, sir. But in some cases also, sir, in leaf and root, also same endophyte we have found. That's why we tried another leaf or root or stem. Sir, I can understand. Yes, sir. Sir, I yes, sir. We have also tried the leaf and root of the Oryza burti. We have found in some cases that same endophytic fungus. Only that 7 endophytic fungus we didn't found. Already we have found 27 endophytic fungus, sir. But in some dendrophilus species, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir. I can. Yes, sir. Yes, sir. 3 gels. Sir, actually, uh, we have referred some paper, research paper, that is endophytic specific primer. Okay, endophytic specific, when we tried in endophytic specific primer, so all paper, especially they have mentioned the when the endophytic fungus amplified, the region is 550 to 650 or 700 base pair. So only this region, firstly, I have tried in ITS, TEF and RPB. So, sir, yes, ma'am. This photo I didn't taken, but the endophytic specific primer we have tried, and all the sequence, all the amplifying region is 550 to 650. Yes, ma'am. Ma'am, different enzymatic activities, growth promotion capacity, and when we tried in dual culture assay, also these two species have higher antagonistic activities towards the rice diseases. Yes, ma'am. Yes, sir. I can understand, sir. Four pathogens, sir. No, no, sir. These two endophytic fungus are showing higher antagonistic activities in four diseases. Yes, sir. Thank you, sir.
Yes, sir. She's online. She's online, but she's not audible. I'm asking her to restart the meeting. Just ask her to just tell that once in the mic she will be able to.
Hello, do you hear me? I cannot hear her. How can you say whether she is hearing me or not?
She's able to hear you, but her voice.
Okay. If her voice is not coming, then tell her to wait for another 20 minutes. In the meantime, she can fix the problems in her end, and we'll go for the next presentation. Tell her that. Okay, let her fix her problems. Yeah. So next speaker will be Akhta. Yeah.
Good afternoon, everyone. Good afternoon. I'm Ekta Bhattacharya. I'm pursuing my PhD from Indian Statistical Institute in the Agriculture and Ecological Research Unit under the supervision of Shuparna Mandil Vishwas ma'am. So I'm here to present work which I had done entitled Malic and L-tartaric acids as new anti-sprouting agents for potatoes during storage in comparison to other efficient sprout suppressants. We have to change, right? Okay. Yeah, it's a PDF. That's why. Okay, actually, yeah. I wanted it to be safer side, but then it's a problem right now. Okay, whatever. Now, a potato is the fourth most important food crop after rice, wheat, and maize. And it is eaten worldwide. And it is being the first and the most important non-grain food crop worldwide. The worldwide production of potato exceeds 300 million metric tons. And because its wide distribution and high yields, it is considered a very important food crop for this food security in the face of this growing population. In India, actually worldwide, India ranks second in potato production, data given by FAO in 2016. And within India, West Bengal ranks second after Uttar Pradesh in potato production, given by the data in 2018 and 19. Now, potato is considered a semi-perishable good as it has high natural moisture content. So the growers have to sell their yields as soon as possible after the harvesting due to lack of their storage facilities and therefore fresh potatoes are available in India for hardly a few months. So good storage facilities or better or cheap. No, it's okay. I'll tell. Okay, just wait. So it's better to, for better post handling and post harvest management is very important for ensuring the supply of potato throughout the year. So, change. Yeah. So what is sprouting and why is it a problem? Now, sprouting of potatoes is the germination and the growth of the eyes of the potato, and it is the significant factor that contributes towards the low shelf life of the potato tubers. After sprouting, actually, the main problems that arise is firstly the weight loss due to increased respiration and transpiration. The weight of the potato tubers are rapidly lost, and secondly, the quality degradation is another problem. Increased metabolic activity and remobilization of the stored nutrients cause the degradation of the quality of potato tubers. And thirdly, the toxin production. After sprouting, potato tubers actually produce different types of toxins like alpha-solanines and alpha-chaconines. They are very harmful to human consumption because they have been proven to cause nausea, vomiting, diarrhea, etc., and other harmful diseases. So, to combat that, there are many commercially used anti-sprouting agents. Among them, malic hydrazide and CIPC are very common. But the problem is, recently, CIPC and malic hydrazide both have been reported to have very harmful effects for human consumptions. Actually, CIPC and malic hydrazide both render the sprouting by actually interfering the spindle formation and inhibiting the cell divisions. So there is a chance of bioaccumulation in case of human consumption, and there are reports that this may lead to hindering the other cellular metabolic processes in human as well. And the other compounds that are used as anti-sprouting agents, these compounds are patented compounds. These have been reported from very early 1980s or something. But the problem here are these natural compounds have their other demerits. Firstly, they are very expensive. And secondly, there is a large-scale unavailability of these compounds. And thirdly, the application procedure of these compounds is very costly and labor extensive. Yeah, so actually there is another problem. The thing is, fresh potatoes do not germinate immediately after harvesting. They are, due to their high moisture content, they are immediately transferred to cold storage facilities where they are kept, and when they are released after a certain period of time, most of the tubers emerge from the dormancy and they germinate within 4 to 6 weeks. Now, we actually focused our study to find new sprout suppressing agents that would be cheap, safe, easy to use, and would actually target to hinder the sprouting at least for 4 weeks or even more. So for that, we formulated our objectives like selection of compounds to be investigated for the anti-sprouting abilities, screening of the screening experiments were done for the selected compounds for the sprout inhibition capabilities. Sprout inhibition assays were done on potato tubers for the selected compounds, assessment for the quality parameters for the treated and untreated potato tubers, and formulation of the tentative mechanism for germination inhibition. Now, for the preliminary screening experiments, we selected few compounds which had prior data of some inhibitory activities. Actually, many of these compounds like tartaric acid, mesotartaric acid, gallic acid, caffeic acid, salicylic acid have been reported as allelopathic compounds. Like they have been reported in other unrelated researches that they had inhibition, they had performed good inhibitory effects on germination of model plants, maybe rice or allium or whatever they have used, Arabidopsis, etc. Now we actually searched for these compounds, and we also made it a point to find compounds that they were structurally similar to some of the reported inhibitory compounds like CIPC, that caffeic acid, gallic acid, and hydroxybenzoic acids. These compounds have structurally similar with CIPC. So we made it a point to find out and select these type of compounds. Now for the preliminary screening experiments, we used tubers, and all other experiments used tubers that had emerged from the dormancy periods. And we treated these compounds with a single concentration of all the selected these preliminary screening experiment. And we treated them, and we considered a control set which was untreated. And we considered those compounds as potentially anti-sprouting that could hinder the sprouting of their treated compounds with compared to the untreated ones, and till all the eyes of the untreated tubers had sprouted. So we selected those compounds that could withhold sprouting till that period. So in our preliminary screening, we could see malic acid and L+ tartaric acid had performed the best. So we continued our detailed experiments with these two compounds. Okay, next. So we actually lost so much time now in this thing. So the sprouting inhibition experiments was done like this. Different concentrations of the compounds were tested from 0.1 to 0.3, and the tubers after treatment was stored for 42 days, and the data was recorded at 6 days. Now the quality assessment parameters were considered. Firstly, the loss of fresh weight, then the reducing sugar content, total protein content, and total phenolic content of the potato tubers were tested. Now the results. Now the 42 days long sprouting inhibition experiment gave really encouraging results. As you can see in the figure, the control sets had germinated, all the eyes have sprouted, and the visual integrity and the potato tubers have lost very much weight and they have shrinked as compared to the treated ones. See, the L-tartaric acid and malic acid treated tubers look very fresh, and they have minimal sprouting of the eyes. And in case of data also, we could see at 42 days, the control sets had germinated about 98%, while the L-tartaric acid and malic acid treated potatoes had only 7.8 or 11% of germination, while 13 and 20% above germination. For the quality parameters also, we could see very good results, as loss of fresh weight was minimal in case of the treated potatoes as compared to the untreated ones. The reducing sugar content was also significantly differed in case of untreated ones with the treated ones. Total protein content, however, had a very unexplainable profile, because L-tartaric acid treated ones was significantly different from the other treated and untreated ones, while total phenolic content had no significant differences among the treated as well as untreated potato tubers. Now, for the tentative mechanism, as we had seen in the literature, there is no specific pathway that is been dealt or that has been reported. There are different kinds of pathways, different thousands or hundreds of enzymes have been reported, but the problem was that there was no single line or flowchart type of working mechanism that was reported in case of sprouting inhibition, even with the reported ones, reported inhibitory compounds even. So now we, as a tentative mechanism, had figured that the malic acid had the best anti-sprouting activity. Now we also tested the fumaric acid. As you can see, malic acid and fumaric acid are actually cis and trans isomers of the same backbone compound. So with that cue, we actually hypothesized that there might be a receptor or an enzyme kind of a molecule which interacts with the cis configuration of this molecule and thus undergoes the inhibitory reaction. While in case of the trans configuration, that is the fumaric acid, it does not fit that typical receptor or enzyme sites and the inhibitory reaction does not occur. So the takeaway messages, firstly, anti-sprouting compounds are the need of the hour to combat post-harvest losses, and organic alternatives are the future for post-harvest handling and storage. Malic and L-tartaric acid have shown promising sprout suppression, and the compounds did not show any quality degradation due to storage, and these two compounds may prove to be very useful for small-scale vendors as they have to store and market mostly after the potato is being released from the cold storage facilities. This study has already been published in 2021 by our team. So I would acknowledge my supervisor, my scientific advisor, Professor Panchanan Pramanik, my lab mates, and I would like to convey my gratitude to AgriVision 2022. Thank you. Actually, this was actually a real long experiment because the corona happened between this. So we failed to test this treatments on different types of varieties, which I aspire to do that. Kufri Jyoti. Yeah, that is the most commonly used and the Kufri Jyoti has the main germination problem, at least it is very commonly available, and even in markets, in some local vendors do, when we choose and go to the markets, we can see the Kufri Jyoti variety is already sprouted. Actually, total phenolics was mainly a quality parameter, as we have studied in the patent and all. They actually consider it as a parameter of quality of potato tubers, along with reducing sugar and, Okay. Actually, okay, but actually I came across studies where actually they were happy that the potato tubers were having. No, we didn't find any difference. That was a very, and when we actually. Okay. 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 Hmm. Okay. Okay. No, no, no. We actually only tried the local, that is the consumable. Mm, yes, there are. We have. I don't remember the names, but yes, there are. Oh. Oh, Indian species. Oh, that's way. Huh. Okay, fine. I get it now. I have no idea right now.
I'm asking you, sir. process is having that same difficulty.
Hello, can you hear me? Hello.
Oh, yes, yes, you are audible.
Okay. So, shall I share my
Yeah,
screen?
yeah. You can share your slide and go for presentations immediately.
Okay, actually the host disable participant screen sharing. Please enable the participant screen sharing.
Yeah, it's done. It's done.
Okay, okay. Thank you. Thank you so much.
Okay. Yeah. You have 10 minutes for presentation and then we will have a discussion.
Yeah, sure. So, good afternoon, everyone. Today I'm going to present my research work, which is titled Evaluating the Potential Role of Natural Products from Pleomyce in Urea's Inhibition and Nitrogen Utilization for Enhancing Soil Health, a rhizosphere manipulation strategy. Fertilizers are essential for the growth and development of crop plants. Based on nutrient content, fertilizers are classified into straight fertilizers, that is when they provide a single nutrient, and multi-nutrient fertilizers when they provide more than two nutrients. Plants require nitrogen as a primary nutrient in large amount compared to potassium and phosphorus as seen in the global consumption of agricultural fertilizers. Nitrogen is so vital because it's a major component of chlorophyll, amino acid, nucleic acid, as well as component of energy transfer compound. Nitrogen is converted into multiple chemical forms as it circulates in the atmosphere. Plant can absorb only nitrate or ammonium from the soil by their root hairs. If nitrate is absorbed, it is first converted to nitrite and then ammonium for incorporation into amino acid, nucleic acid, and chlorophyll. When a plant or animal dies or an animal expels waste, the initial form of nitrogen is organic. Bacteria or fungi converts this organic nitrogen into ammonia by a process called ammonification or nitrification. The conversion of ammonia to nitrate is performed primarily by the soil-living bacteria and other nitrifying bacteria. Denitrification, which is the reduction of nitrate to nitrogen gas, thus completing the nitrogen cycle. And it is done by bacteria Pseudomonas and Paracoccus. Nitrogen fertilizers take the foremost among all the fertilizers. And as you can see, inorganic nitrogen fertilizers are mainly of four types: nitrate fertilizers, ammonium containing fertilizers, nitrate and ammonium containing fertilizers, and their amide forms. More than 80% of the fertilizer used in this country are made up of nitrogen fertilizers, particularly urea. It is extremely efficient in increasing the production of crops. Urea fertilizer, which is also popularly known as 4600, is a simple and straight fertilizer that provides the major essential element nitrogen in the ammonic form. Besides having high nitrogen content, it has other benefits as well, such as low cost of manufacturing, ease of handling, storage and distribution, high solubility in water, and reduced corrosivity. Once urea fertilizer applied to the soil, it is hydrolyzed rapidly by the enzyme urease in two stages. In the first stage, ammonium carbonate is produced, which rapidly and spontaneously hydrolyzes into ammonia and carbonic acid. The active site of the enzyme urease contains two nickel atoms with interatomic distance of about 3.5 angstrom. And urea binds in a bidentate manner to its via carbonyl oxygen atom to nickel atom 1 and amino group to nickel atom 2. Now, this soil applied nitrogen is used by the crop plants for the growth and development, but at the same time, a significant amount of this applied nitrogen is lost from the soil through various mechanisms, which include volatilization or urea hydrolysis, leaching, which is the movement of nitrate through a soil profile, denitrification, which is when the soil organism uses nitrate as an electron acceptor instead of oxygen and converts it to nitrous oxide, dinitrous oxide, and nitrogen gas, which is released into the atmosphere. This huge loss of nitrogen can be checked by using urease inhibitors. The inhibitors can stop a substrate from entering the enzyme's active site and thereby hinder the enzyme catalyzing its reaction. Some of the compounds listed here are tested commercial inhibitors of urease, but most of the commercial inhibitors of urease are non-degradable. They are costly and unfriendly to the environment. So the potential plant-derived natural product as urease inhibitors can be valuable for the development of improved nitrogen fertilizer formulations to increase the food production. Some of the groups of natural inhibitors are listed over here, like terpenoids, phenolics, flavonoids, xanthones, coumarins, alkaloid, glucosinolates. These glucosinolates are biologically active sulfur-rich compounds that are found in the Brassicaceae family of plants, such as cabbage, broccoli, cauliflower, etc. And this glucosinolate compound, it is hydrolyzed by the enzyme myrosinase to produce an unstable aglycone and a beta-D-glucose molecule. So we proceeded with the following objectives. First is to study and investigate new plant sources that can effectively and specifically undergo urease inhibition, followed by collection of this sample and preparation of the extract based on solvent polarity. Next, to study the fractions ability to inhibit urease inhibition, and also to understand the mechanism of inhibition by the Lineweaver-Burke kinetic analysis technique. Then we analyzed the compound, the bioactive compound, by HR-LCMS analysis. And finally, we did the compound isolation and study with molecular docking to ascertain the binding energy and mode of interaction of this isolated compound with the active site of urease. So for our present study, we have chosen the Cleome gynandra as a tested plant species, which is also known as cat's whiskers, is an erect branched annual herb. It is highly nutritious vegetable and also contain a high amount of glucosinolate compound, also possesses numerous medicinal values. Yet this plant is considered as a neglected and underutilized weed species in most of the world. So plant samples were collected and categorized into 4 groups: whole plant, stem, leaf, and root, and they were air dried, and then it was grounded into fine powder using sample miller machine. And this 200 grams of the powder was soaked in 500 ml methanol and was vortexed in a mechanical stirrer for 2 hours at 3000 RPM. It was then filtered through sintered disc funnel filter and concentrated in a rotary vacuum evaporator. This concentrated crude extract was fractionated using solvents of various polarities, starting from non-polar to polar, and it was purified using a column chromatography and thin layer chromatography. These purified samples were then stored in airtight bottles for further study. So urease activity of the crude extract of Cleome gynandra whole plant leaves and stems were determined by measuring the ammonia released during the reaction at 640 nanometer using phosphate buffer solution of pH 8.2. Urease enzyme concentration was 0.03 mg per ml. Test compounds 500 to 1000 ppm. Urea concentration from 500, 1000, 1500 and 2000 micromole was used, and incubation time was maintained at 20 minutes. And thiourea was used as a standard. The percent inhibition was calculated by the following formula. And the concentration of the inhibitor required for achieving 50% inhibition of the enzyme was determined by monitoring the inhibition effect at various concentration of the sample using different substrate concentration compared to the standard thiourea. At all substrate concentration, it's seen that in case of leaf and stem, there is a significant difference in the isotopic value with respect to the thiourea. But in case of whole plant, due to synergistic effect, the inhibitory activity is not significantly different from that of thiourea. Now, to understand the mechanism of inhibition, here we did the Lineweaver-Burke plot or the double reciprocal plot, which is a graphical representation of the Lineweaver-Burke equation. It can distinguish competitive, non-competitive, and uncompetitive inhibition. The various mode of inhibition are compared to the uninhibited reaction. Here we have plotted 1 by V versus 1 by S for different concentration of the crude whole plant extract to identify the kinetic parameters Km and Vmax. The kinetic parameters also indicated a non-competitive mode of inhibition, but the Km did not vary significantly and the Vmax varied significantly with the increase in concentration of the whole plant extract. Similar results are observed in case of leaf and stem. Since the whole plant crude extract showed maximum inhibitory activity, so we were interested to analyze the compound present in the whole plant extract. Each LCMS spectra of the whole plant extract of Cleome gynandra showed 8 sharp peaks with the following retention time, and the following 8 new compounds were identified. So to study the interaction of the compound, molecular docking study was performed using Autodock Vienna software based on the published structure of Jagbin urease. PDB ID used here is 3LF4. And generated dot complexes were analyzed based on the binding energy values and the bonding interaction patterns. These figures were created using PyMol. Surface representation of the active site flap of the Jagbin urease with glucocapurine shown at the entrance of the binding pocket. And this glucocaprin showed strong hydrogen bond interaction with the amino acid residues aspartate 730 and lysine 716 with interatomic distance of about 6.74 angstrom and 4.14 angstrom respectively in the active site mobile flap of Jagbin urease. Interestingly, it was found that the hydroxyl group of the glucocaprin was affected the molecular conformation of the Jagbin urease, which enhanced the binding affinity with this amino acid residues and hence resulted in lowered enzymatic activity. Similarly, we have also done the docking with flutic acid and propionate, and we have shown the surface representation at the entrance of the binding pocket. Similarly, flutic acid and propionate showed strong hydrogen bond van der Waals and pi alkyl interaction with the amino acid arginine 639. And this observation also is soundly supportive of the involvement of the active site sulfhydryl group in the inhibition process. So coming to my conclusion, the neglected and underutilized cleomgynandra showed potent anti-Jagbin urease activity, which can be used for the development of new agricultural formulations and integrate them with agronomic practices for reducing nitrogen loss and also to increasing the nitrogen uses efficiency. The kinetic parameters here also indicated a non-competitive mode of inhibition where the Km value did not decrease significantly and Vmax value is decreased as the crude extract concentration is increased. So next, we also found 8 new compounds from our HRSMS study of the purified whole plant extract, and the interaction of the compound was studied through molecular docking, and we found specific interaction with amino acid that were responsible for lowering the enzymatic activity. And of course, this compounds needs to be analyzed further for development in agriculture and therapeutics. These are some of my publications and awards. I would like to acknowledge my guide, Dr. Suparna Mandal Bishwas. I am extremely grateful to Dr. Panchanan Pramanik, our research advisor. And lastly, I would like to thank my lab mates and other members of my unit. Thank you, and also thank you to Agrivision for giving me this opportunity to speak in front of so many respectable audiences. Thank you.
Can you please go to your Lineweaver bark plot?
Yes, sir.
Okay, you say this one is non-competitive inhibition, right?
Non-competitive.
Inhibition, right?
Yes, sir.
And have you measured at what concentration it actually caused significant decrease in the velocity? Inhibitor. What is the Ki, inhibitor constant for this particular substance?
Inhibitor constant is, I have forgotten to written, it was, I need to check. I couldn't mention over here. I need to check that.
Yeah, you need to have measurement of inhibitor constant.
Okay, sir.
Because inhibitor constant is the indicator of the capacity of an inhibitor.
Okay, sir. Okay, sir.
Okay. Then my question is, how could you measure the IC50?
Okay, I measured the at different. I use different concentration of my substrate, and I found that at what substrate concentration it is inhibiting the enzyme, and it is 50% inhibiting the enzyme.
And you got a concentration where exactly 50% inhibition occurred? Because IC50 is not a experimental concentration. Nobody can determine IC50 from experiment. IC50 is always a statistical concentration. You will have to apply a particular statistics to calculate the IC50 from the experimental data. Right?
Yes, right, sir.
So then how could you calculate this IC50 without applying any specific statistical method?
I actually did here ANOVA, but
IC50 is 50% inhibition of the activity. So inhibitory concentration 50%. Okay.
Yes, yes, sir.
And never in the experiment you will get a concentration where exactly 50% inhibition will occur. Therefore, you will have to apply, you will have to take multiple data points. From the slope, you will have to determine the equation. And
Yes,
from
sir.
the equation,
I did that, sir. From the slope I determined from the equation y equals to mx plus c.
And what was the slope? What was the parameter for the slope? What was the measuring parameter?
Measuring parameter, the concentration of urea and absorbance.
Concentration of urea is the substrate.
Yes.
So what is the measuring parameter in this case? What did you measure? And from that parameter, you determined ICP.
Yes, I measured the absorbance at 640 nanometer. I measured the ammonia released at 640 nanometer.
Okay. And then?
And then I plotted the graph in Excel sheet in absorbance versus urea concentration.
It was linear.
Yes, sir.
It was linear. The relationship was linear.
Yes, sir.
Okay, but normal kinetics, enzyme kinetics says that the equation cannot be linear.
Oh no, it was not linear. It was high. It was not linear.
Okay, tell me what is the application of your research?
Yes, sir. I can tell. Basically, I am searching for urease inhibitor to reduce the nitrogen loss by the enzyme urease, because when we apply urea fertilizer to soil, the urease enzyme rapidly hydrolyzes it and it produces ammonia, which is lost as a nitrogen in the environment. So we'll reduce the nitrogen loss, and this could enhance the nitrogen uses efficiency, and also we can increase our food production via this, and by using minimum amount of fertilizer.
Okay, thank you very much. It was a nice presentation, and wish you all the best for your future research.
Thank you, sir. Thank you so much.
Good afternoon, everyone. My name is Srimoy Mitra, and I am here to present a part of my PhD work titled Isolation of Halophilic Bacteria from the Agricultural Soils of South 24 Parganas in Indian Sundarbans. I am currently pursuing PhD under the supervision of Professor Pabitra Bonik at the Agriculture and Ecological Research Unit of Indian Statistical Institute, Kolkata campus. India, as we know, is an ecologically diverse country. It sees its diversity in its flora, fauna, and the soil also is not an exception. It sees a variety of soils, including saline soils. Now, it can be seen here, but I did mention the parts of India where saline soils have been found, like in West Bengal, in parts of Uttar Pradesh, Punjab, Andhra Pradesh, Tamil Nadu, Rajasthan, Gujarat, etc. Now, I am here to talk about the state that I come from, West Bengal. And Sundarbans, as we know, is a World Heritage Site, and it also holds one of the largest mangrove ecosystem of the world. Approximately, it covers an area of 10,000 square kilometer and is being shared within two countries, Bangladesh and India. Bangladesh holds 60% of the of the Sundarbans, and India holds 40% of it. Now, talking about halophiles or halophilic or halotolerant bacteria that I had mentioned in my title slide. What are these? These microorganisms are capable of surviving in saline and hypersaline conditions. What makes them so special is that they have this quality to influx the salt ions outside their cell by taking in certain low molecular weight organic molecules like ectoin and hydroxyectoin. They replace those molecules with the salt ions so that the sodium and potassium ions had to be driven out of the cell. And that is how they maintain the osmotic pressure inside the cell and stays within the saline conditions. These microorganisms have already been reported in certain parts of the Indian Sundarbans, Maharashtra, Rajasthan, Tamil Nadu, etc. Now, talking of halophiles in agriculture, they have immense importance. As I had mentioned here, they have importance in nitrogen fixation, phosphate solubilization. They act as plant growth promoters. They also can hydrolyze urea. They can mitigate salt, calcium and magnesium solubilization, biodegradative enzymes, mitigation of pathogenic diseases in plants, especially crop plants, and formation of nanoparticles. Now, my objectives were firstly to collect soil samples from the agricultural soils, because when we talk of Sundarbans, we immediately go to the mangroves because it's very interesting. But I am here to take samples from the agricultural soils and then to isolate halophilic or halotolerant bacteria from those soil samples, and lastly to check for or assess their biofertilizing abilities by making them grow in different media, that is to check their capability for nitrogen fixation, phosphate solubilization and potassium solubilization. So firstly, I collected soils from 6 different locations. I collected 18 soil samples. The soil samples were then subjected to serial dilution with physiologically saline soil in 1:10 ratio. Then, from the dilutions -3, -5, and -7, the inoculum were taken and were plated in nutrient agar plates, which were already enriched with 2%, 5%, and 10% of sodium chloride concentrations. The ones which showed promising growth in the 10% concentration were further streaked in 20% concentration just to check how much they can tolerate. Then, by doing this, I got 23 halophilic bacteria or bacterial isolates from the 18 soil samples. Then I grew them. Since I was only using NaCl, I needed to check whether they can grow in other soils also. So I grew the isolates in 2 other salts, calcium chloride and magnesium sulfate, to check whether they can survive. Further, the isolates were grown in Alexandrov, Pikovskaya and Jensen's media for checking their capabilities. And for the soil, I checked its pH and electrical conductivity. These are my sampling locations in the Sundarbans. I collected soils from Kagdeep. I collected soils from Kagdeep, Pathor Pratima, Namkhana, Bokkhali. These are certain pictures of the sampling locations. Not following my command. It's progressing. Anyways, this is the data of CFU per ml only for the colonies that grew in the 10% plates. These are the pictures of the growth of the bacteria in the different concentrations of NaCl. These are the pictures of the streaks that I did in the 20% NaCl. I got these 23 isolates. Then in the calcium chloride and magnesium sulfate, 2 of the isolates among the 23 grew in both of them. Rest showed individual growth, and some of them did not grow at all in those 2 soils. Staining behavior, by doing gram staining, it was seen that most of these isolates were gram negative. Few of them were gram positive as well. These are the pictures of the isolates, of the 23 isolates that I got. Now, when I introduce the nitrogen fixation, phosphorus solubilizing, and potassium solubilizing media, only one isolate that is highlighted here showed growth in all these 3 media. Few of them grew on them individually, but only one of them showed promising growth in all the 3 media. These are the pictures of the isolates in the Pikovskaya, Jensen's, and Alexandra media. These are the data of the soil pH and electrical conductivity. Now, to conclude, I would say that till now I have got 23 halophilic bacterial isolates. Among them, only one showed promising nitrogen fixing, potassium solubilizing, and phosphorus solubilizing capability. 2 of them grew in calcium chloride and magnesium sulfate. Hence, as I mentioned before, this is a part of my PhD work, so I will continue this work and will study for their field applications as biofertilizer. So I will also explore more and more halophilic or halotolerant bacteria in this field so that it can be used as a biofertilizer, so that we can replace these as the immensely used chemical fertilizers in those regions that the farmers use there to combat the saline soils or the adverse conditions they face there. And I feel that by lessening the use of chemical fertilizer is a way of doing sustainable agriculture for a better tomorrow. These are the person I would like to acknowledge. Firstly, my supervisor, Professor Pabitra Bonik. Next, I would like to extend my gratitude towards AgriVision 2022 for giving me this opportunity to present my work. I would like to thank Dr. Shutripta Sarkar, the Dean of Studies and faculty members of ISI Kolkata, for providing me with lab facilities. I would like to thank my lab members, my family and friends for their help and encouragement. These are the references that I've used. And thank you for your patience.
So thank you. Nice presentation. It's not audible properly here. Am I audible?
Yes,
Okay.
sir.
Okay. So my question, I have very small queries. One of them say, why have you taken calcium chloride and magnesium sulfate? Because normally in the oceanic system, sodium chloride salinity determines the level of salinity.
So, but it is not the only salt that is present.
So what is the contribution of calcium chloride and magnesium sulfate then together to salinity?
Sir, exactly that I cannot tell you now. I would surely look into it, but they do contribute.
Literature, if you go through
Yes,
literature, it
yes.
is approximately, both of them together is approximately 15%.
Okay.
So therefore, I don't think calcium chloride and magnesium sulfate tolerance is a necessity for your study. One, and you told you have started with halophilic / halotolerant. Are they same?
No, sir.
Then why halotolerant?
Sir, I am, as I mentioned, this is an ongoing work. I don't know whether I will get an halotolerant. These are not halotolerant. Halotolerant organisms are those which can grow in non-saline conditions also. But halophilic needs salt to grow. So I am not sure whether I will get any halotolerance in future. So I just have put that on slide.
If there is no salt at all, will the bacteria grow?
Yes. No, it needs a little bit of salt.
Sodium is a necessity.
Yes, sodium chloride is necessary.
Sodium is a necessity for bacterial growth. So my question, another question is, you tried up to 10% and even more.
Yes.
Does it exist in nature, such a salinity?
No, but in for extreme conditions like what Indian Sundarbans have been facing, these tropical cyclones. When I went to those sampling locations, what I heard from the farmers is that it gets immensely salty, so that they cannot cultivate for like 1 or 2 years. So they lose that time. So it might not exist, but I just wanted to see whether they can grow in lower concentrations also, but they can also tolerate that much.
Yeah, you see, when the soil is extremely salty, it contains about 500 micromole or more than that of sodium chloride. And when you calculate 500 micromole, it comes around sodium chloride together 500 micromoles comes around 3%. So high salinity that is existing in the soil is around 3%, excepting the coral reefs and hypersaline conditions where you find much evaporation.
Yes, yes, sir.
So going to 10%, I don't think is a necessity for you to
Okay.
determine the halo tolerant or
Okay, sir.
halophilic type.
Okay, sir. I'll remember.
Yeah, yeah.
Yes, sir. I intend to do so. It might not replace them completely. It will replace them to a certain extent because they have to use a large amount of chemical fertilizer to retain the fertility in the land because it gets very saline. In other saline parts also, not just Sundarbans. Yes, sir. So, but I don't know, but I'm just saying that these are indigenous species. They are present there. Yes, yes, of course, of course.
They are in a particular
Yes,
association,
yes.
they are in a particular density. If you make invasion of other species, automatically it will disturb the balance.
Yes, sir. I will, when I will look into the field application of it, I will make sure that I can minimize that effect so that I can use it in an amount which doesn't hamper the ecosystem that is present there already. Thank you, sir. Yes, sir. Thank you, sir. I will definitely remember this. Thank you.
Is there any more question? Then I must thank the speaker and I will request the next speaker,
Thank you.
Madhusmita,
Thank you, sir. Thank you,
to
ma'am.
make the presentation. Madhurima, sorry, Madhurima Datta. You have any problem? Okay. You are slide Pachitara? Then Prachi, you come, you make the presentation in the meantime. You make Prachi's presentation, yeah. And then next one will be from Madhurima.
Hello. Science doesn't have to be beautiful, it has to be effective. A very good afternoon to all. Myself, Prachitara Raut, working at NRRI Cuttack. I'm going to present the topic haploid doubling in antherculture of elite indica rice hybrid CRHR32. Now, rice is one of the most important crop. Rice is one of the most important crop, highly nutritious, and it's one of the rich in carbohydrate resource in the world. It can be the rice will be definitely will become 25 to 30% more in. Okay. Okay. Rice is one of the most important staple food, and it is for important for the malnourished and poor people in the world. So, but the rice production has to be increased by 2050. It will be more 25 to 30% more. So we have to find the way for the rice production more because of the decreasing land due to industrialization and due to various climate changes. So we went for the rice hybrid after this rice sale becomes static after the green revolution. So we went for the rice hybrid production. Which is giving more than 50 to 20% higher than the inbreds. So this is the CRH32 in my topic, which is a release variety from the NRRI Cuttack. So, but rice hybrid is have also some of the constraints due to exorbitant high seed costs, troublesome for farmers to procure new seed every year. There's unpredictable environmental conditions and the quality of the produce, which has hindered the production of the rice production in the India. So we went for the antherculture. Then what is antherculture? Antherculture or androgenesis is one of the most important biotechnological mechanism. The underlying principle is that we have to stop the development of pollen growth, and it makes due to the embryophytic route to find a complete haploid plant. So DHS produced through androgenesis can shorten the breeding cycle, fix agronomic traits in homozygous trait, and enhance the selection efficiency of useful recessive agronomic traits. Now, why we are taking why we have if we have traditional breeding purposes, then why we are taking the DH technology? As we can see in the slide, through conventional breeding, to get a pure homozygous line, we get 6 to 8 generations, which is a very long time, and it can also dissipation of unique gene combination over long course of segregation generations. While in double haploid technology, in one single generation, we can get a homozygous plant. But already there are so many reports on Japonica rice in the antherculture. We are getting it is amenable to antherculture, but there are limitations in the indica rice. That is, it is recalcitrant to tissue culture, anther necrosis, and the most frustrating problem is the albino shoot regeneration. Now, I can take a quick look on the pioneers of the antherculture. That is, I've given 3 photographs. That is Black Silly, Guha, and Maheshwari. Black Silly is one of the pathfinder to get in produce a natural haploid in the Dactyla stramonium, while Guha and Maheshwari first in 1964 reported the direct development of embryo from microspores of Dactyla inoxia, while in rice it was first haploids are produced by Nijeki and Ano. Now, what are the advantages of antherculture? It is very simple method, less time consuming, it is very responsive, used in cryogenic studies, and it has profound utility in the crop and improvement. The DH applications are the development of improved rice cultivars, gene stacking, reverse breeding, transgenic research, cutter mapping, mutation studies, and accelerating plant breeding coupled with the marker assisted selection. So for this study, although we have reports on antherculture indica, japonica, and the intersubspecies hybrids, but reports are meager on the production of double haploid from the release hybrid varieties to antherculture. So this is one of the methodology. First we collected the boots, then we went for the microspore stage determination, then we go for the cold pre-treatment, we calculate the green callus induction and green albino plant regeneration. We transferred to plants in the net house and ploidy assessment was done. Then we went for morpho-agronomic characterization, molecular validations, and lastly we for grain and nutrition quality analysis. Some of the factors which affect the rice androgenesis are physiology of dormant plants, development stage of the anthers, pre-treatment, osmotic stress, culture medium, and genotype. Now, these are the results. We have found that anthers of the pollen of the anthers at the mid-nucleus stage gave the appropriate result. So these are the photos we can say where the anthers are first dusted, then it turns brown, swollen, and then we can here get the calli here protruding from the anthers. Then the calli which were 2 to 3 millimeter, we transferred to the regeneration media, that is MS regeneration media, and we got the plantlets, green plantlets. After 10 to 12 days, we found the green spots. Then we found after 20 to 30 days, the green plantlets are formed. Here I got an albino plant also. After that, we transferred to the net house transfer. Then there was ploidy assessment in the net house. That we can see it was diploids, polyploids, and haploids. Diploids were characteristics by the normal morphology, while the polyploids were the leaves are very large and thick, and there was spikelet fertility is also very less, while haploid is characterized by it has a very small stature and with no spikelet fertility. Then we were in the diploids. In the next slide, I will tell about the distinguish of the how we distinguish the diploids and double haploids, the molecular markers. I'll go to the next slide then. But this is the morphoagronomic characterization of the 150 DHs in the field conditions. After that, when basis on the 2 seasons, we field a trial for 2 seasons, and basis of the morphoagronomic characterization for 2 seasons, we got 20 selected lines for the replicated trials. Out of 20, we got 6 plants, 6 DHS plants, which gave higher yield than the parent CRHO32. So as I said, out of the 3 basal media, N6, MS, and SK1, I got result on the N6 on 2nd day, followed by the 8th day, that is 16 for the callus induction, that is 16.35. Without any hormones, we didn't find any callus inductions, and with the increase of the hormones concentration, we found the callus induction has decreased. In the regeneration media, we found the callus induced from N6 has given better result, that is 71.66% in MS media. Now, how this is the 150 DHS parameters, the plant height, morphoagronomic parameter, that is plant height, DFF, flag length, the panicle length, spike length. Out of the 150 DHS, I have compared with the CRHA32, and we have seen that some are better and some are lower than the DHS. The replicated trials based on 2 seasons also, we found 6 DHS which are giving higher than the CRHA32, that is 2 to 5.16% more than the yield than the parent. Now, I have told how to distinguish between the diploid and double haploid, which cannot be done through the flow cytometry because they have same chromosome number. So here, through molecular markers, this is a heterozygous. The CRHA32 hybrid is showing the heterozygous bands and the diploid. Through 4 markers, I have concluded that this is the one diploid line, which out of 151, which we took out from the field, and we went for the 150 DHS. Parental allelic distribution in DHS showing 1:1 out of 650 markers, 40 markers, so polymorphism between the parent, that is CRHA32, or that is CRL, FF5 and CRMS31A, out of which it is showing 1:1 distribution. We saw that they are not repetitive and not from the same gametes of the CRHA32. These are the 40 SSR markers which are showing in the pictorial diagram, showing the various positions in the chromosomes. And I said grain and nutrition quality. In this malnourished world sustainable age, protein and iron, zinc, and phytic acid are one of the most important, not phytic acid, protein, iron, and zinc, one of the most nutrition cards. In rice, it should be less of iron is and protein and zinc is less, while the phytic acid concentration is more. Phytic acid inhibits the concentration of iron and zinc. So in this analysis, we have found that 1 DH I got to found, which has same yield than 6 tons per hectare, but it has high protein, high iron and zinc, but low phytic acid. Yes, sir. So these are the silent findings of my experiment. That is, we got 2-day incubation at 10 degrees centigrade, giving 16.35% callus induction. Highest regeneration, soot regeneration was 71.66%. Out of 150 DHs, 20 were found to be the promising, which gave grain yield at part higher than the parent, CH32. 6 DHs gave higher grain yield than the parent, that is 2 to 5.16% higher, and 1 DS for high zinc, iron, protein, and low phytate. DH technology could be taken as an efficient method for among all the developed speed breeding techniques for development of promising rice lines to feed the increasing populations. It could be utilized for the production of the biofortified rice in a short period of time. Thank you.
How many
Yes, sir.
have been released as DH to the farmers?
Sir, 2 have released Satyakeshna and Falguni from BHB 71 and KRH 22. 2 DH varieties in India, it's about NRRA, but many varieties are about
From NRM.
NRRA, sir, but in many, in China, there's many varieties in release.
How can you check this albino problem?
We are on the verge of, but I cannot disclose here, but we are on the verge of the albino problem.
And also you have shown in one slide that all of them have more yield than the parent. Okay. Most of them are.
6.
Look, so what can be the reason? Just because of anthroculture?
It could be the dominance effect, but it could be the dominance effect or epistatic and environmental effect, or it could be gene expression, which will be going on in this season. After I complete this, I'm again back. And sir has given some experiments to check whether we have only checked the grain yield, but other parameters will be checking in this. Yes, sir. Yes, sir. Yes, sir. The marker assisted breeding, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Pure lines, sir, with different genes combinations, sir. And this project which I have at SRF, that also we are doing, sir. Thank you.
Hello.
Good afternoon all. Today I am going to talk about one of my research topic that is entitled here as Identification and Metabolomic Characterization of 2 new pigment producing post-harvest fungi from Indian gooseberry. In agriculture, post-harvest handling is the stage of crop production immediately following harvest. In short, it includes cleaning, sorting, cooling, and packing. So we can say that the post-harvest handling largely determines the final quality of any crop. In 2015, ICAR and CIPHET together showed in their report that is the post-harvest losses was maximum in fruits and vegetables among any other crop. And they estimated the loss is around 4.5 to 15.8% per year. And this loss is mostly causes by the post-harvest infections. The pathogenic microbes are mainly responsible for this post-harvest infections, and fungal diseases are prevalent among them. Though the pre-harvest application of different fungicides has reduced the post-harvest infections as well as increased crop shelf life. A large number of natural compounds has been reported to get from the microbial world after plant, and fungi signify themselves as the most prolific producers of these bioactive compounds with 42% of the total. As the fungal genome contains multitude gene clusters of secondary metabolites, so their bioactive compounds has unique structural diversification than the synthetic chemical compounds. And some bioactive compounds has potential activities such as aflatoxin from aspergillus and fumonisin and fusarium is both are the toxins we get from the microbial world, but the penicillin from the penicillium species is the most well-known used as drug. So the beside different bioactive compounds, we get some pigments from the microbial world as well. And they are used in edibles, especially in oriental foods, for centuries. And these foods are mainly consumed by the people of southern China, Southeast Asia, and Japan. But these pigments are still forbidden in many other countries. Some of the pigments known to possess important remedial properties like anti-cancer, antimicrobial, anti-mutagenic, anti-obesity, etc. But some of them are known to possess or reported to possess some mycotoxin which is undesirable from them. Phyllanthus embolica, colloquially known as Indian gooseberry in English and amla in Hindi, is a plant of Phyllanthus family, distributed largely in tropical and subtropical areas, but it is originally native from India. It is the most acknowledged herb in the famous known medicinal literature, Charak Samhita. The fruit of Phyllanthus embolica is highly acidic with ascorbic acid content 20 times higher than that of an orange. But they have higher spoilage rate and shorter shelf life. In our present study, we get to pigment producing fungal species from the rotten fruit surface of them, and these are the new addition to the microflora of amla. These are some of my objectives, that is isolation and molecular characterization of fungal isolates, metabolomic assessments and screening through bioactivities of the isolated secondary metabolites, understanding the relevant mechanism adopted by them to grow and colonize in a high acidic environment of the fruit, exploring purified bioactive materials from these fungal sources after removal of mycotoxins in biotechnological applications. So we collected the rotten fruits from the commercial market near Damdam, Kolkata, and we brought them into the laboratory into sterilized polythene bags for microbial isolation. And depending on the pigment production, we name them as the red pigment producers as the PERF and the yellow pigment producers as the PEGYF. After 7 days of inoculation, we observed nearly circular colony in white mycelium growth with red rosy pigment diffusing in the PDA plates from the PEIRF isolates. After another 7 days, we observed the white mycelium growth turns into grayish green colony with velvety texture with a good amount of red monosense pigments. Under microscope, we observed n mass of conidia attached to conidiophore structure. On the other hand, in the yellow pigment producers, we observe nearly circular colony with white mycelium growth with the crutosulcate structure with little amount of yellow pigment. For another 7 days, we observe that is the mycelium growth turns into dark greenish in color with a good amount of yellow pigment on the PDA plates. Under microscope, we observed abundant number of spores with septate hyphal structure. To confirm the identity of the fungal species, we have isolated the total genomic DNA and we amplified and sequenced on the basis of ITS region and BAN-A gene. From that, and the sequence submitted to the NCBI Gene Bank, and here we can see the accession numbers of them. By the phylogenetic analysis, we confirm the species, the red pigment producers, that is PERF, as the Talaromyces atroroseus, and the yellow pigment producers as the PEGYF, that is the Penicillium coryndensis. For ITS region analysis, we using the neighbor joining method, and we used 1000 bootstrap analysis, and from that we can conclude the genus of the red pigment producers as Talaromyces and the yellow pigment producers are from the Penicillium. But we cannot conclude that these are Talaromyces atroroseus or the yellow pigment producers as the Penicillium choriospondaitis. For that, we studied the coding gene, that is the beta-tubulin A gene, and we use the maximum likelihood method for that. And from this phylogenetic tree, we can confirm that the Talaromyces atroroseus are the red pigment producers and the yellow pigment producers as the Penicillium choriospondaitis. Further, we want to analyze the secondary metabolites they synthesize in the PDA plates. So we isolated the pigmented media after removal of the mycelium growth in an extraction flux with an unique solvent mixtures of ethyl acetate, acetone, and methanol, and vortex them by using mechanical stirrer, and subsequently filtered them through the synthetics funnel, and we get the collected pigmented material, and we concentrated them into the rotary vacuum evaporator, and then the crude material were weighed and passed through biochemical tests like total phenolics, total flavonoids, total tannins, etc. And the antioxidant activities, we performed DPPH test and the ABTS test. From the analysis, we get that we only get the total phenolic test is the successful, other flavonoids or the tannins is absent from the both isolates. And we can see that Penicillium choriospondaitis has better phenolic content than Talaromyces atroroseus. Similarly, when we perform the DPPH and ABTS test, we get that Penicillium choriospondaitis show the better antioxidant activity than Talaromyces atroroseus, and these are their IC50 values. Further, we want to analyze what are the secondary metabolites actually present in them. So for that, we fractionate the crude material through the heliotropic series of solvents. Methanol fraction was selected on the basis of the biochemical activities, and the resultant methanol fraction materials are purified through the column chromatography, and the sample is will go to analysis for the HRLCMS further. So my significance findings are, this is the first report of the occurrence of red pigment producing Talaromyces atroroseus and yellow pigment producing Penicillium choriospondaitis on Indian gooseberry from India. In addition to antioxidant activities, Talaromyces atroroseus secretes large amounts of monoscous red pigments, which can be used as food colorant or as industrial dyes, while the Penicillium choriospondaitis produce a good amount of yellow pigment, which needs to be characterized properly further. These bioactive compounds, along with the pigmentous secondary metabolites of isolated fungi, need to be further molecular characterization for depth knowledge regarding their potential for commercial use. And by following necessary methods for complete removal of mycotoxins, purified bioactive materials from these fungal sources can be explored in biotechnologically merchandised products. These are some references I have been gone through for my present study. I would like to convey my sincere gratitude to my principal investigator, Dr. Suparna Mondol Bishwas. I would like to thank our head and professional in charge for their enormous laboratory facilities. I would like to thank the member of aggravation to let me showcase my work here. Lastly, I would like to thank my lab mates from ADRU Department of Indian Statistical Institute of Kolkata. Thank you for your patience.
Yeah, that was good. But I think you must go for this characterization. You're saying that it can be like a possible dye or something, but you need to perform some experiments for that. Exactly how can you do that? Okay. Have you thought of anything?
Yes, that's why I put this last slide. For the further detailed secondary metabolized characterization, I will go to the fraction of the samples, pigmented samples through the allotropic series. Then we will do the HRLCMS to identify what compounds are present and then exactly. Or those compounds has any bioactivities or not, or any mitochondrial present in that.
As of now, you have not done anything.
No,
That's
no, no.
okay.
It is
Thank you.
under process.
All the best.
Thank you.
Thank you all for your nice presentations and wish you all the best. Wish you all the best for your future research. Now we'll break for the lunch and we'll move to the Department of Botany.
hello. Ouch. What? Good afternoon to all. I would be talking regarding the novel antibiotic-based selection for developing transgenic plants. As we know, there are next slide. Yes. So we know there are various methods for developing transgenic rice, different plants, and basically agrobacterium mediated transformation and other sort of transformation procedures are there. We understand the methodology for developing transgenic line. So now we are in the era of CRISPR-Cas mediated genome editing process technology which can develop transgenic lines. So this particular technology being widely accepted because of this side-directed nuclease. It can change a one base pair, or you can mutate it at our point of target. So, but it has some limitations, very few limitations, because if you would like to transform with a novel transgene taking from other sources, then this particular technology may not work. For that, you have to go for this SDN3 type of transformation, like where you can develop transgenic line with help of a marker genes. So the marker genes is important for cloning. The marker gene is important for developing plants for the selection. That marker gene, for the help of this marker gene, we can select, transform, and regenerate the transgenic plants. So there are a few negative points. I mean, adverse effect of transgenic. It is not accepted under the biosafety regulations, because it has some adverse effects, like it can horizontally transform. It can have the effect on the gut microflora that can transform to a next generation which may not have any work. So it is recommended to remove the selectable marker genes, so that it would not come under this biosafety regulation. So question is how to remove this the selectable marker genes. First thing is it is necessary to develop for developing transgenic line. It is necessary to utilize the selectable marker genes. So how to remove, that is the question. There are various technology for the removal of this selectable marker genes. The selectable marker genes will have no purpose after selection of transformants. So it is important to remove the selectable marker genes as well as these reporter genes. So there are many reporters which people does use in developing transgenic lines. So in order to remove, or in order to develop transgenic line without any selectable marker genes, there are many approach which we will see. These are the non-antibiotic or herbicide resistance genes if we can have in that vector. Other ways are excitation of this resistance marker genes through this co-transformation, site-specific recombination, intrachromosomal recombination, and transposon technology, etc. So various herbicide resistance genes that has been already people used for developing transgenic genes, but again these herbicide resistance genes would be difficult for removal in the transformants. So we can see there are many other way of developing transgenic lines without having these marker genes. For example, we can see there are co-transformation method where you can transform your vector in two different strains. One is the first one, this two different strains, or you can transform the two vector carrying one your selectable marker genes, another one is uh, your gene of interest, and clone in a single strain of agrobacterium and transform into the plant cell. The other one, third one is the vector containing the selectable marker genes along with the gene of interest and transform into the plant cell. So after analysis, so through this segregation analysis, we can find out which of the transformants is having your gene of targets, and the other transformants which are having the marker genes that can be eliminated, that can be removable. So for that process to find out which are the genes having your gene of interest, then that may needs a few generation, or it may require 2 to 3 generation minimum to develop this marker-free transgenic lines. But this is a technology which works, which people have already demonstrated. They have developed transgenic lines with this technology, but it may require hard effort or laborious techniques. So for that, you have to go for the co-transformation process. We have to go for positive transformation, positive selection. After positive selection, you may get the lines with having the positive selection marker, negative selection marker, and the target gene. And finally, you have to go for the negative selection to find out the gene, the plant having the gene of target. Another site-specific recombination you must have heard. I'm not going to the detail regarding developing transgenic lines through the recombination process. You can use the Krelux P or FLP, FRT, and RS, this recombination mediated marker deletion. So is to develop these marker-free transgenic lines. So another method is using help of transposons. As you know, the transposons are the jumping gene which you can clone the gene of interest in one vector. Then with the process of the crossing, then it can segregate to another plants, and finally you have to find out the gene of where the gene of interest is present in which plant, and you can finally segregate them out. So for that, we need to have the specific method protocols for developing transgenic lines that we have standardized and published in 2011. It's a very renowned protocol having 178 number of citations. People have used successfully this methodology for developing transgenic rice. So that if we use this protocol for developing transgenic rice, we can use for either of this technology. Besides this technology, we have used some other physiological selection by which we can develop these transgenic lines. For example, we have used this methyl glycol system where we have used 2 glycylase enzyme overexpressed, and how we develop this transgenic line without any marker or without any selectable marker genes that we will discuss. For that, we have published this report in earlier for manipulation of glycylase pathway that confers the tolerance in rice. For that, we have taken these 2 genes of methyl glycol. So these are the common techniques which we use for developing transgenic lines. These are the 2 enzymes. So with help of these 2 enzymes, the methyl glycol can be detoxified very easily with the overexpression of glycylase 1 and 2. So we successfully developed transgenic line and validated in using different technology like PCR Western, Southern, and many other tools. We successfully see they tolerate salinity stress and drought stress. So these are not coming. So this has been again shown in different essay by using this fluorescent dye, which suggested or again confirmed the ability of transgenic line for less accumulation of the sodium ion. So these are the 2D analysis which has been performed. Just I wanted to show you how this glycolase system work, how we can develop this transgenic line without the any selectable markers genes, for example, hygromycin, etc. So here we can see how this pathway works. So in one of the your pathway of glycolase, you can see DHAP, dihydroxyacetone phosphate, forms the methyl glycol. The methyl glycol is a toxic which it avails if it is present in more than its basic or basal level. So for successful degradation of methyl glycol, we have two enzymes, glycolase 1 and 2. So methyl glycol is converted to SLG, S-lactoyl glutathione, by the presence of glycolase 1 enzymes. The SLG again convert to lactic acid in presence of glycolase 2. So in presence of stress, we find the methyl glycol overexpresses. So if we can degrade the toxic methyl glycol in presence of glycolase 1 and 2 enzymes, so we overexpress glycolase 1 and 2 enzymes in plants, and we could see the methyl glycol level is successfully degrading and the toxic effect is not visible. So how it has been done, we have taken this glycolase 1 and 2 enzymes, clone in vector, transform into agrobacterium transformation. So we have taken this methyl glycol system for possible selection of a module for raising the transgenic lines, recently published in 2021. So for understanding what concentration of methyl glycol is lethal for the plant cell, we have taken different concentration of methyl glycol. As I said, methyl glycol concentration, if it is high, it will be toxic for this cell. You should know the methyl glycol is present in uniformly in every kind of cells, starting from the bacterial cell to eukaryotic to prokaryotic cells. So we have taken the different concentration of methyl glycol. We can find at certain concentration let's say for plant cell, for callus it being lethal, and the LD50 and the lethal dose has been defined for the callus as well as the seedlings. So we have taken the LD50 concentration about 3 mg per liter for raising transgenic lines. So you can see there is no selectable marker genes in the vector, only glycolase 1 and 2 are there. Another construct has been taken. So here you have glycolase 1 and 2 along with the HPT2 gene. So the HPT2 gene is your hygromycin resistance genes. So we have taken two different construct, transform into the calli of the rice. We have successfully generate the transgenic line with the help of these both the construct, having glycolase enzymes with hygromycin and having glycolase enzymes genes without hygromycin. So we developed the transgenic lines and compare what is the percentage of transformation efficiency in if you select in presence of Mg methyl glycol, and if you don't select, if you select with the presence of hygromycin. So it is here important to understand that in the selection we have exogenously provided the methyl glycol and a dose about LD50 dose about 3 mg per liter. So we could see it has equally good kind of transformation efficiency like HPT2. So this has been validated through these molecular techniques by using PCR, southern, western, and different essay are being done with understand whether the transgenic line with the glycoxylase genes having glycoxylase 1 activity or not glycoxylase, it could found the transgenic lines produces or do have this glycoxylase 1 and 2 activity. So from this data we can understand uh, the marker-free transgenic line which are selected on presence of these methyl could uh, able to develop the transgenic line, so they could able to grow in presence of these methyl. So so here this is up to one technology where we have developed the transgenic line with selection of methyl instead of this HPT2 or hygromycin genes. So another technology we have used by taking the trihalose phosphate synthase, trihalose phosphate phosphatase gene. So these two genes have been taken. Here, this is the pathway how this sucrose has been converted to glucose 6-phosphate, and how this glucose 6-phosphate is converted to trihalose 6-phosphate in presence of trihalose phosphate synthase, and how this trihalose 6-phosphate is converted to trihalose in presence of trihalose phosphate phosphatase. So trihalose 6-phosphate synthase and trihalose 6-phosphate phosphatase, these are two genes have been fused and clone in a vector and develop and transform into the agrobacterium followed by the plant cell. So this is the agrobacterium mediated transformation in rice cali, taking the trihalose phosphate phosphatase and trihalose phosphate synthase genes. So the production is selected not in the presence of any of the selectable marker gene. This is selected in presence of high concentration of glucose. So the transformants which having the trihalose phosphate synthase TPS and TPP genes, they could able to grow in presence of high glucose concentration. So we could successfully raid the transgenic line in by selecting on this physiological selection of glucose. So after then we have been this has been validated through these molecular techniques of PCR southern as well as your western technology. This is the PCR analysis. We have designed specific site primers for the plants, the plants having the trihalose phosphate synthase gene, having both trihalose phosphate and synthase and trihalose phosphate phosphatase gene. This has been again done the southern analysis, western analysis, and for the presence of these lines. So individual transformants TA7, A18, X19 was found. So this has been again revalidated for the abiotic stress analysis. As we know, trihalose is present in resorption plant. As a resorption plant, for example, Silajanana, one is a resorption plant we found in many of the cases of Balijatra and a few other plants. If we put this plant in water, within 3 hours of putting in water, it again rejuvenate and becomes green. So this is because of the presence of this trihalose. It has been already established. So we did this analysis, and we could found the trihalose overexpressing line, which has been selected through the presence of the glucose. They are marker free and they could able to provide stress tolerance for various stresses. It has been examined. So that's all. Thank you. Thank you very much. I'd be happy to discuss any sort of questions if you have. Okay, thank you. If you have no question, we can move to the next one. Thank you.
Good afternoon, all. Myself, Dr. Devana, scientist at ICR NRRA Cuttack. Today's my presentation is on PF54 and its orthologs, their role in effective management of rice blast disease. So this is the rice with various applications, which most of them are consumables. So rice is grown worldwide, and it is feeding almost 1/3 of the global population. So among many diseases which affect rice, rice blast disease, which is caused by Magnoporte varizae, that is a hemibiotic fungus. It is very deadly disease, considered one of the most dangerous disease in case of rice. The annual loss caused by this disease, it is sufficient even to feed almost 60% of the 60 million people each year. Deployment of R genes, that is a resistance gene, it is considered as one of the most effective and eco-friendly approach. A lack of broad spectrum resistance and continued evolution of the AVR genes is one of the main constraints for durable resistance. So for that, we have to keep on identifying new genes. Among the rice germplasm, wild rice is one of the most important source of resistance genes. So in this presentation, I will be largely talking about developing rice lines for the, blast resistance using transgenic approach. So initially in 2013, we evaluated many wild as well as land races, that is different germplasm of rice. And in that we found that among others, Vareza rhizomatis, latifolia and officinalis were giving complete resistance. And in this we had different controls, resistance as well as susceptible. So to start with, the first study, it was about map-based cloning of rice blast resistance gene PR54, which was earlier called as PR54RH. And subsequently, after our allele mining study, its ortholog was studied from Vareza rhizomatis, and it was named as PR54RH. And in our study, we have cloned one more ortholog, that is PR54OF from Vareza officinalis. Initially, we wanted to know whether this gene is expressed and what is the phenotypic response of the Vareza officinalis against Magnoporte. This is wild rice, first A, B is the after infection leaf, C is the susceptible control. So through expression analysis, we found that PR54 ortholog gene is expressed in officinalis and it is being induced after 12 hours of the infection. Otherwise, it has basic expression even at 0 hours. So compared to officinalis, when we compare it with PR54 from Tetep and PR54RH from rhizomatis, you can see here at 0, 24, 48 hours, there is no expression of, in case of PR54. In rhizomatis also, there is no expression up to 48 hours. At 72 hours, in both cases, there is a slight increase in PR54 and there is a increase in at 96 hours only. So there is a change in the pattern of expression. In case of Vareza officinalis, it is suddenly induced at 12 hours. That means very early stage of infection. Then we went for cloning. These are the various stages of cloning. We finally cloned 2.1 kb complete gene. And these are the different structural aspects of, first is susceptible gene from type I309. Next is its ortholog from TETEP, and C is rhizomatous, and D is from officinalis. So among the three orthologs, officinalis gene is biggest and smallest is from TETEP. So through domain analysis, we found that there is a variation in the domains also, and PI54OF has one unique domain among the three, that is STI1, that is stress inducible protein 1, which is binding to the RAC1 and heat shock protein, which are component of defensome complex. Then we did some in silico analysis just to find whether they are forming any specific structure, and most of you may be knowing most of the typical R genes, they form horseshoes like structure. So similarly, PF54, RH and OF, they could form typical horseshoe like structure, and from susceptible one, the last panel down one, it is not a complete structure. Then through structural stability, we found that PF54OF protein, it is highly stable under native conditions, indicating that it has a proper structure, 3D structure compared to other orthologs. Then we did docking experiment. Through docking, we found that all the resistance orthologs of PF54, they interact with AVR gene. This is the last one, AVR PF54. This is the AVR counterpart. Whereas there was no interaction between susceptible allele and AVR. Then we further studied what are the amino acids involved in the R-AVR interaction. You can see in PF54, CC, non-LRR and LRR domains are involved. Whereas in case of PF54 rhizomatous, only non-LRR and LRR are involved. In case of officinalis, only non-LRR is involved. This indicates that only, that means all the three orthologs have different amino acids and domains involved while interacting with the AVR counterpart. Then we performed subcellular localization of the R proteins. The first, this is the PF54, this is again, this is officinalis. PF54 is localized in all the parts, that means membrane, cytoplasm, and this rhizomatous is largely found in the membrane. You can see here the first panel, whereas officinalis is found largely in cytoplasm. So to go for transformation, that means to develop a transgenic rice, we cloned the PF54OF. Otherwise, PF54 and PF54RH were already used for rice transformation. Then this is a transformation, various stages of transformation, and these are the final transgenic, putative transgenic plants. Various analysis like PCR, then southern were performed just to confirm that these putative plants are actually transgenics. Then we did phenotyping, that is a phenotypic assay. Among the 5 transgenic plants, 4 were indeed, among the 6, 5 were indeed having resistance, whereas one number, that is number 5, it was showing susceptible. That may be attributed to site of gene integration. Then the same construct was used for transformation of Japonica rice, that is type I309. The earlier one was Indica rice, that is Pusa Sugandh 2. Then we also performed molecular analysis to confirm the integration of the transgene through PCR and CAPS assay. And this is the bio assay for the Magnoporthe resistance. Then we predicted a model. Through that, what we can say is, after the insertion, that means formation of the aprosorium, it releases the AVR protein that is recognized by the non-LRR domain, that is in case of PR54OF. Then STI1 protein will bind to the STI1 domain, which recruits other components of the defensome. So till now, it is inactive complex. There is one protein, that is rho-GEF. It activates the total complex through GTP. Then it leads to this on top one, H2O2 mediated hypersensitive response. Then through next stage, through MAP kinase, it leads to effector triggered immunity. That means, I think you all know, AVR PF54 is the effector. The resistance triggered by this is called ETI, effector triggered immunity. So we further pyramided PF54 and its orthologs, one in Indica, another one in Japonica background. Coming to the applications of PF54, till now there are various studies. PF54 gene has been deployed all over the globe. The yellow lines are the other than India, and rest all are India. In China, Malaysia, Switzerland. So this PF54 has been deployed for the development of rice lines for blast resistance. In summary, PF54 is the largest among the three orthologs with a special STI1 domain. PF54 is early inducible compared to the two other orthologs, means it has immediate effect on the it leads to immediate resistance response, higher HR response. Resistance mechanism of PF54, that means all the PF54 is somewhat different because of different domains. Then they show overlapping pattern of resistance, means all the three orthologs, they show different level of resistance against different strains of the Magnoporthe. Then PF54 is deployed in different rice lines. I have shown that table. It is deployed globally, and there is more scope for the further deployment, because till now we have used only PF54. We can explore rhizomates as well as officinalis, though it is a little tricky because specifically officinalis tetraploid rice. We have to break that barrier and bring it to diploid background, and we can use further for blast resistance. My acknowledgements are to my chairman, Dr. T.R. Sharma, then our lab mates, seniors. Thank you. Any questions?
Hello, am I audible? Hello. 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 Good afternoon to one and all. My name is Shraddha Jamwal, and I have submitted my PhD degree under Dr. A.K. Mohanty. And now currently I am working as a PI under the Biotechnology in Ignition grant, which is granted by the BIRAC. And today I'm here presenting a part of my PhD research work titled TMT-based proteomic approach revealing key players of pre-implantation and endometrium receptivity during embryo and endometrial epithelial interactions. So first I will describe about the implantation. Embryo implantation is a critical step for establishing a successful pregnancy, and implantation takes place in different steps such as hatching of blastocyst, elongation, attachment, and cell fusion. So during the implantation period, that is like 20 days of implantation periods, embryo remains loosely bound to uterine cells and communicate with uterus so that it can receive embryo for the implantation. The implantation is a critical window of pregnancy, and it has been reported that about 80% of embryo die in this period, and which has a major impact on the economy process in dairy farming. So there are various factors which affect the embryonic mortality, like genetic factors, endocrine factors, nutrition factors, heat stress, unfavorable uterine environment, and palpation effect. So in this study, we had tried to understand the molecular basis of uterine cells, which takes place during the interaction between embryo and uterine epithelial cells by using high throughput proteomic approach. So in the methodology which we have used in this research, we had isolated and cultured and characterized fellow endometrium epithelial cells, and these cells were then used for the proteomic study. These cells were first incubated with and without blastocysts, and protein was extracted from these two samples and digested using trypsin enzyme. And these digested proteins were then labeled with the different TMT labels, and these labeled peptides were then pooled and desalted and analyzed in LC-MS. The generated raw data was then was analyzed using Proteome Discoverer software against Bostore's proteome database. The identified proteins were then analyzed using different bioinformatic tools such as Flugo and String. So these are the results which we have obtained in this experiment. This is the images showing the buffalo endometrial epithelial cells. We were able to obtain more than 95% endometrial epithelial cells in its purity, and In proteomic study, we have obtained total number of 4,130 proteins, out of which 69 were differentially expressed in endometrial epithelial cells in the presence of blastocysts when compared to normal endometrial epithelial cells. And out of 69, 24 were upregulated and 45 were downregulated. So this is the list of my differentially expressed proteins which were identified in the experiment. And this is the heat map showing the protein abundance between the two groups. See, so this blue color is showing the low abundance protein, and the red color is indicating the high protein abundance in the sample. These differentially expressed proteins were then classified on the basis of its biological process, cellular component, and molecular function by using Klugo software. And this is the visual, this network was visualized in Cytoscape software. And here we can see that these differentially expressed proteins were enriched in different processes like apoptosis, cadherin binding, disordered domain specific binding activity, substrate adhesion, dependent cell spreading, glycolysis, gluconeogenesis, chromatin silencing, etc. So an interactome was generated by using Spring software for the differentially expressed proteins, and the confidence score was kept at 0.7 for the generation of the network. And the network was then enriched for different pathways, and the string analysis showed that that the proteins were involved in 5 different pathways, such as invasion of epithelial cells and ECM receptor interaction, focal adhesion, glycolysis, gluconeogenesis, and actin cytoskeleton regulatory pathways. So this is the schematic representation, which is showing what are the molecular changes taking place inside the endometrial epithelial cells during the embryo and epithelial interaction. So when blastocysts come in the close proximity of endometrial epithelial cells, it releases some factors which then bind and activate different pathways like AKT, 3K pathway and STAT3 pathway, which ultimately regulate the expression of different genes like peroxyredoxin, cyclophilin, integrin. And these are the proteins which were identified in our study, and the expression of these protein was higher in the study. So this is the list of some key player proteins which were identified in the experiment, like filamin A, azirin, cyclophilin, peroxyredoxin, alpha-actinin 4, tropomyosin alpha 4. And the other study has been done by other researchers, and they have shown that these molecules are very important for implantation, and any downregulation of these protein expression leads to implantation failure. So I will conclude my slides that implantation is a very important and multi-step process which requires precisely timed molecular mechanism and different biological processes. And using this proteomic technique, we were able to get insight to understand the molecular basis of implantation. And the study showed that early embryonic signaling is important to guarantee a successful establishment of a pregnancy. And the endometrial epithelial cells were enriched in adhesion molecules, were enriched in important molecules which are responsible for endometrium receptivity, like adhesion molecules, cytoskeleton regulatory proteins, focal adhesion proteins, antioxidant molecules, inflammatory stimulant molecules, signaling regulatory molecules, which collectively function to regulate cell adhesion, signal transduction, and ultimately control the embryo implantations. So we suggest that these proteins can be used as a potential target for improvement of uterine environment, so that we can enhance the fertility in the dairy animals. However, as the implantation is a complex mechanism, this research area still required detailed, in-depth research, and therefore it is a necessity to develop a standardized research method to understand the molecular basis of uterine endometrium in the early stage of pregnancy. So in acknowledgement, I would like to thank my advisors, Dr. A.K. Mohanty and Dr. Sudarshan Kumar, and my institution, National Dairy Research Institution, for providing me the facility to carry out the research, and the funding agency, National Agri Culture Science Fund, and BIRAG for providing the funds, and Department of Biotechnology for providing the computational facility under the framework of BTI S network. And I also like to thank AgriVision 2022 for providing the platform for representing my research. And this is the list of references. Thank you.
Good afternoon to one and all present here. So first of all, I'm Parameshwaran. I'm working as a scientist in Crop Improvement Division of National Rice Research Institute, Cuttack. So my work, I think many Parameshwarans are here. So my work is on working on genome editing and say other data analysis part like machine learning. So I thought since this AgriVision, that the main objective is on doubling the yield and income of farmers, I thought I may discuss few points on how we can use machine learning and the genome editing for increasing the yield. But in this case, I'm not talking about yield of rice in India. So I'm not talking about yield of rice in India, but I want to concentrate only on yield of rice in Odisha, especially Odisha, this state. So before going into my presentation, first of all, I would like, please excuse me because I could not prepare it properly. So what I thought, I will be discussing some case studies. Say, let us take 2 case studies. So of how we can use machine learning and genome editing for increasing the yield. So if you are expecting some basics of say machine learning or genome editing, I don't have the slides on that. So I'm just going to discuss 2 case studies. So my first case study will be on understanding the rice production in India. Let me. Can you move the slides from there? Yeah. So I hope you all know some basic information on rice production. So are we still a rice deficient country, or we are producing a sufficient amount of rice? So really, what is happening recently, we are really a rice sufficient country. We are producing the rice which is more than the demand which we need. That means in this year, our rice production was around say 120 million tons, and the demand is only around 109 million tons. So generally it means the recent increase in rice production is really a big success story in India. This is not only rice, there is also an example of wheat. So both the cases, our food production have crossed 300 million tons. That is because of increase in rice production as well as wheat production. So, so you can see here actually the rice production. No, this data is up to 2018, and you can see the rice production is around 12 million tons, and rice yield is also around 2.6 to 2.8 million tons. So my first question here I would like to ask here is, really do we want to increase the rice production? Because we have sufficient rice production in our country. So really we want to target improving the rice production. So this is my first question. So another point to substantiate my question that really we need to improve rice production is, you can see the bar chart here. There are, say, rice production is divided into different zones in India. Say central zone, eastern, southern, northwestern zone. And when you see here, the eastern zone, which comprised of, say, West Bengal, Odisha, Bihar, eastern parts of Uttar Pradesh, they are the recent drivers. They are the recent drivers of rice production in India. This is not the case 15 years back. In 15 years back, you might know it, especially it is central zone and then southern zone. These 2 zones were really important in improving the rice production. But recently, in 10 to 15 years, there is a change in, there is a shift in rice production status in India, eastern states, especially eastern states where rice production is really high. That is why we are seeing increased rice production, which is more than the demand of our country, and also we are exporting say sufficient quantity of rice to the world. But actually what happens, even though eastern region of India, they are the drivers of rice production, especially the yield of rice in Odisha is relatively less. So we have to understand that the yield of rice in Odisha is relatively less when compared to, say, Uttar Pradesh, when compared to eastern parts of Uttar Pradesh, Bihar, West Bengal. So in a real sense, if we want to increase yield of rice, it indirectly means we have to concentrate on improving the yield of rice, especially in Odisha. So that is the point I want to make here, that the regional dynamics are shifting. In case of rice production, we have to concentrate more on Odisha. But there are several risks. I mean, there are several factors that reduces the yield of rice in Odisha. But still, if I say that we have to double the income, or we have to double the yield, especially we have to concentrate on rice yield in Odisha. So in this scenario, friends, just I want to say summarize, improving the yield in Odisha is a major objective. So first of all, we have to know what is the factor that is increasing the rice production, especially in eastern parts of India, whether area is increasing, whether say fertilizer inputs are increasing, or whether say farmers are using high yielding varieties. So first of all, we have to understand, so why eastern parts of India are now the leaders, they are the drivers of rice production in our country. In this case study, I have used the machine learning approaches to identify why the rice yield is more, especially in recent years, say 10 to 15 years of rice production. I think you might all know really what is machine learning. Machine learning is training the data using computer algorithms and testing the data for its accuracy. In simple terms, machine learning is we will train the data and we will generate a model, and the trained model will be used for predicting or testing our data. So one of the major prominent point of machine learning is feature extraction, which means which among the input variables are really important. In our case, let us take an example. So here I have given area, production, irrigated areas, fertilizer usage, seed production, and these are 5 different factors which we have taken. So recently in rice, so we have taken on, say, production, irrigated area, seed distribution, fertilizer, and also manures. So we don't know what is the exact reason for the increase in rice production. So for that, I have used a machine learning approach, and one particular approach is artificial neural networks. So this artificial neural network, it functions similar to our neural response. So it takes the data, it trains the data, and finally it identifies the major factor that is responsible for yield improvement. So in this case, area, production, irrigated area, seed distribution, fertilizers, and manures are used, and we have developed a model, and the error rate is also 0.00348. That indicates the model has a very less error rate. One of the advantages of machine learning is that error rate will be very less. And in the next case, I have also used random forest machine learning models to identify the variants that are important. So generally, this random forest models that try to analyze the data in terms of variables, and from the variables, we will be able to find out which one is really an important variable. So here you can see among the different factors like production, year, seed distribution, fertilizer, manure, irrigated area, and area, the seed distribution apart from production. So production is a, say, positive variable here. So we are trying to understand why production is more in recent years, and year is one of the factors. But among the inputs, say seeds, fertilizers, and manures, you can see seed distribution is the prominent factor that is responsible for increase of rice yield in recent years. So this is how we understand that. So seed distribution is really important for yield improvement. So seed distribution here we means the supply of quality seeds, especially foundation and certified seeds to the farmers, and that is responsible for the increase in rice production. So once we know what are all the factors that is important for rice production, the next approach will be just to increase the seed quality seed distribution percentage in Eastern India. So just if you all know that seed replacement ratio is not say optimum in case of rice, especially in Odisha. So if we try to improve the seed replacement ratio with the usage of say certified seeds, that will really help in improving the yield of Eastern India, especially Odisha. So this is the summary of this work, where we have used different factors and we have found that quality or certified seed distribution is prime factor in increasing the rice yield in recent years in India. So this can be targeted especially in Odisha to improve the rice yield. So this is the first part of my case study. So wherein we have seen how machine learning can be used to improve the rice yield. But supply of quality seeds, it depends on the management aspects, it depends on the resources and infrastructure. But still we have to improve the yield of the popular varieties, because most of the varieties farmers are growing now, they are say old varieties which is released say last 10 years, 15 years back, especially Swarna, the popular variety in Eastern India that is released say 20 years back. So all these things makes a point that not only that seed supply or seed distribution is really important, we should also try to improve the yield of the popular varieties. In this case, NRRI is also working on genome editing. Here we have used one particular gene, IPA1, ideal plant architecture gene, to improve the yield of one popular variety, say Swarna. So friends, you have to understand here, this ideal plant architecture is a major gene in rice. So it is involved, it is most of the nearly 90% of hybrids in rice has the superior alleles of ideal plant architecture, especially in China. So Chinese breeding actually revolves around this IPA1, DEP1, GN1A, like 3-4 major genes. So we thought we will edit the gene. One particular advantage of editing IPA1 is different alleles have a different phenotype. For example, a stronger allele will show very less tillers but increase in number of spikelets per panicle. Weaker allele will show increase in panicle numbers. So this IPA1 is really interesting gene where different alleles of the gene have a different effect on the parent, on the varieties. So then we have used say CRISPR based strategy, genome editing strategy, and we have developed the say T2 plants or M2 plants, and you can see this is the architecture of this IPA1 edited Swarna lines. So the first one is the parent variety, this is the parent variety, and all these are the edited lines. Since genome editing, we are using transferred DNA through agrobacterium mediated transformation. We have also identified the Cas9 free plants. So Cas9 free plants are one which don't have any vector imprints in it. So once we have identified this Cas9 free plants, and then once the lines were developed, we tried to characterize, we tried to do molecular characterization of these edited lines through sequencing. And this is the chromatogram, and one particular site, we have targeted this microRNA 156 binding site because that regulates the yield in rice. So this is the chromatogram based validation of genome editing, and once you have validated at molecular level, now we have to characterize these plants at say phenotype level. Especially this gene increases the number of spikelets, tillers, many other aspects. And this is one example of an edited line where when compared to Swarna, the parent variety and also popular variety, this edited lines shows increase in number of panicle branches, number of spikelets, say panicle length. So this is one of the major advantage of genome editing, where you can change gene, you can change the nucleotide sequences of a gene to have an increased yield. And we have also measured quantitatively what is the benefit of these genome editing, genome edited lines of IPA1. So here you can see the number of spikelets, say per panicle. It is around 180 or 160 in parent variety Swarna, and edited lines, they are showing more than say 200 spikelets per panicle. And also number of panicle branches. So it is around 10 in parent variety Swarna, whereas edited lines shows more than 10 number of panicle branches. And this is the single plant yield. In Swarna, it is around say 30 gram, whereas in edited lines, it is more than say 35 or 40 grams per plant. So from this data, or from this work, we would like to say that we can also use genome editing for increasing the yield of the popular varieties, say like Swarna, MTU 1010, Samba Mashuri, many other varieties can be targeted through genome editing. Not only that, we have edited Swarna variety, some other irrigated varieties also showed the same response. Naveen, another popular variety in Boro ecology, especially in Northeast India, it also showed the same response of increase in number of spikelets per panicle. So let me conclude this talk. So generally, rice yield is important, but in Odisha it is highly important because Odisha's agrarian economy fully depends on rice. So in that also, eastern parts of Odisha has different say climatic factors as compared to western parts, and these eastern parts of Odisha farmers are also highly due to the cyclones and other depressions, the yield of the rice in eastern parts of Odisha is relatively less when compared to western parts, where other irrigated sources also available. So in this talk, I tried to explain you about two different approaches. Say one is machine learning, another one is genome editing. Both can be used to increase the yield. So genome editing, which can be used to increase the yield, whereas machine learning can be used to understand why in recent years, why in Odisha the yield is less, or why in India the rice yield is really high. So that would be my conclusion. So I thank all of you for listening this talk. If there is any queries, I may answer. And thank you then. Thanks for giving this opportunity. Thank you.
Hello, I am Srija Priyadarshini, pursuing MSc Agri Horticulture from Soya University. Now I am going to present my topic. Okay, sir. Okay, sir. Okay, sir. Okay, sir. Thank you, sir. Now, Vigna vexillata, an underutilized tuber crop. So, what is Vigna vexillata? Vigna vexillata, otherwise known as, it is otherwise called as wild cowpea or zombie pea, and it is usually cultivated for its pod, seed, and tuber. Change the slide. Here. The wild cultivars are found in Africa, America, Australia, and Asia. And cultivated only, these are cultivated only some parts of Africa, and it is not found in other continents. Tuber contains its 15% protein, which is more than some other tuber crops. It is adapted in infertile or some adverse climatic conditions like drought, waterlogging conditions. So I am with the importance of this crop with the following objectives. So like these are some of the objectives we are carried out at CTCRI regional session, Bhubaneswar, to study the productivity and profitability of the crop under variable spacing and deblossoming. Then as it is newly introduced crop, so we want to know what is the quality parameters of this crop, so that we know what is the quality of the Vigna vexillata crop. Then third one is we actually know what is the economic importance, whether it is economically viable or not. So this is carried out at CTCRI campus under the genotype IC 259504 using split plot design, where the main plot is spacing, that is 4 different spacing are there. 45 into 15, 45 into 30, 60 into 15, and 60 into 30 centimeter. Where the subplot is deblossoming, where in F1 we taken that inflorescence removal, and where F2 inflorescence retention. Then these are the phenology of the crop. You see, this is the 30 days after sowing, where tuber bulking is small bulking is there. After 45 days, somewhat more bulking is there in tuber. Then after 90 days, you can see S3F1 and S3F2. In S3F1, we didn't see any pod because deblossoming is there in S3F1. So no pod will occur in that S3F1. But in S3F2, we will get two yield, that is pod yield and tuber yield also. Then after 105 days, it is S1F1, you can see that more bulking of tuber is there. And this is the graphical representation of the Vigna vexillata. Effect of spacing on pod and tuber yield. It is yield per plant. Those are yield per plant. First one is S1. You can see here in S4, the spacing 16 to 30 centimeter, we can get more yield per plant. But in S1, that is 45 into 15 centimeter, the yield is very low. So we conclude here that in per plant yield, when we come to per plant yield, the spacing is 16 to 30 centimeter is best for us. In deblossoming, you can see in F1, only tuber yield is there because we remove the flower. So there is no pod yield. But in F2, we get both the yield, that is pod yield and tuber yield. Again, this is the yield per hectare, effect on spacing on pod and tuber yield. Here, somewhat changes of yield because of the due to population, as we taken in hectare. In S1, you can see it is 45 into 15 centimeter. We can get more yield in closer spacing because of the population of the crop. And it is very low in S4, that is 16 to 30 centimeter. But in per plant, it is 16 to 30 centimeter, we get more plant yield, per plant yield. And in effect of deblossoming, you can see F1 will not yield any pod, but in F2 we can get two, pod yield and tuber yield. Another parameters we added here, that is tuber equivalent yield, where it is same thing, that is S1 will get tuber yield and pod yield. So equivalent yield also more here. And in F1 there is very low tuber yield, so equivalent yield also low. In F2 we can get both pod yield and tuber yield, so equivalent yield also more. Here the quality and economics of the tuber crop. We see that in quality parameters, that is starch and sugar content, effect of spacing and deblossoming will not affect any of our quality parameters. However, the starch ranges 16 to 24% and its sugar percentage is less than 1%. And cost of cultivation, gross return, net return, we see that it is more in the spacing 16 to 30 centimeter. And the conclusion, we saw that 60 to 30 centimeter is best for our crop because it is gives gross return more and also benefit cost ratio and also product tuber equivalent yield. And with this conclusion is there, if we grow this crop, we can retain the flower because we can get more productivity. And spacing and deblossoming will not affect any of our starch and sugar content. If we get the more maximum yield, so it is 16 to 15 centimeter we will take for this Vigna vexillata crop. Thank you so much for giving me this opportunity. Thank you. Now you, ma'am? F1 means, ma'am, that is soppler treatment where F1 inflorescence removal, ma'am. That treatment is inflorescence removal. No, ma'am. That is main plot and subplot is there. Then F1, F2 is subplot. F1 is for deblossoming. F2 is for retention. Now this floor is open for the discussion.
So why you are taking this plant?
Sir?
Why you are taking this plant?
Sir,
Vigna?
because it is very underutilized crop. As you saw that it gives in Vigna species, whether it is one yield, we all know it gives only pod yield. But it is a underutilized crop that is gives us two yield, pod yield and tuber yield. So we have taken this crop.
Okay, thank you. Okay, thank you.
Thank you, sir.
Vivekanand Panda. Are you present? Payal Kapoor. No one is there. Then Harish Chandar. Yes, Saroj.
Yes, sir. I'm here, sir. Sir, my screen is not sharing actually.
Yeah, actually you are not visible. Your screen is also not visible. I think Saroj, na Payal Kapoor. Saroj.
Sir, is it sharing?
Yes.
Okay, sir.
Your time is 10 minutes. Huh?
Okay, sir. Am I audible, sir?
Yeah,
Shall I start, sir?
sure.
Good afternoon to all of you. I'm Saroj. I'm a research scholar from School of Economics in Sri Mata Vaishno Devi University, Jammu. My research topic is contract farming and its welfare impact assessment, a case of wheat grower in North India. So 1 minute. There is some issue in my screen.
What happened, Saroj?
Sir, I think there is one issue in my screen. It is not
Clear cursor is moving here and there. We can see. You can make it full screen, na? Yes.
but sir 1 minute.
Right here.
So this is the background of my study. After the implementation of new economic policies and reforms in 1991, the difference between agricultural and non-agricultural production growth rates has increased dramatically. The agricultural sector is still going through severe growth stagnation. It is observed in many instances that low and highly fluctuating farm income drive farmers out of agriculture altogether. Moreover, the conventional marketing channels are unproductive, fragmented with low scale and manifold intermediaries. Inadequate infrastructure, poor price discovery, and policy falsification, which serve as deterrents to the farmers' income. So, in order to protect resourceless farmers from agricultural problems, an effective and farmers-oriented agricultural policy must be established from the sowing of seeds to the post-harvest procedures. In this context, contract farming is supposed to be a better remedy for many agricultural ills like lack of quality inputs and access to better market, long chain of multiple middlemen, ignorance of buyer demands. So what is the contract farming? It is defined as an arrangement for the production and supply of agricultural or horticultural products under an agreement between producers and buyers. The essence of such an arrangement is to deliver a certain types of agricultural commodity at a predefined price and quantity that is required by a known and committed buyer, that is usually a large company. This is the review of literature. In the context of contract farming, both type of literatures are available, positive and negative. Some authors like Miyata et al. 2009, Munon et al. 2009, and some others stated that in underdeveloped economies, contract farming is considered as a better way to diminish production risk for the farmers by providing them improved technology, related farming information, and higher end market for the final product. Contract farming have welfare enhancing effect on growers through the enhancement of yield, productivity, and gross margin, and reduced exposure to market risk. Moreover, farmers who participated in contract farming are wealthier in terms of land and other assets than who are not participated in it. And it also generates overall human labor employment. But on the other hand, many studies interpreted contracting system as a tool for agribusiness organization to exploit growers for their own proceeds. Smallholders are exploited by large agribusiness firms due to the limited bargaining power. They are excluded from contract farming on the basis of asset holding size. Some authors stated that generally farmers find that contracts are biased and imposed strictly. Firms provide poor extension services, overpriced input facilities, pass on the risk to farmers, offer low price of the product, favor large farmers, delay payment, and do not compensate for the natural calamities also. After viewing both aspects, positive and negative, of the contract farming, the objective of the study was finalized. So the present study focuses on evaluating the impact of contract farming in Haryana in terms of whether the contract farming benefits the wheat growers or not. And if it benefiting, then whether the welfare implication of contract farming are scale biased or scale neutral. This is the database and methodology of the study. This study is totally based on the primary data. This present study is conducted in Sirsa and Hisar district of Haryana. Sirsa and Hisar district are selected for the study purpose because these two districts contribute the maximum share in total wheat production in Haryana. A total of 754 wheat farmers surveyed from the whole study area, which includes 323 hours of contract farming and 431 hours of non-contract farming. Here, endogenous switching regression method is used to compare the expected outcome of adopters and non-adopters of contract farming, and also to measure the expected outcome of their hypothetical counterfactual cases. There are two types of hypothetical counterfactual cases. One is when the adopters do not adopt the contract farming, and the second when the non-adopters adopt the contract farming. More specifically, under this method, at the first stage, farmers are supposed to determine whether or not to adopt the contract farming based on the expected outcome of contract farming. At the second stage, two outcome equations are specified that explain the relationship between the outcome and the vector of independent variables, conditional on whether farmers adopt contract farming or not. At the final stage, net income of contract farming adoptions on the adopters, that is called ATT, and the non-adopters, that is called ATU, can be derived. This is the econometric framework of this method. So because of the lack of time, I should skip it. This is the result of mean difference test of farm characteristics of the adopters and non-adopters of contract farming. The gross margin and crop yield, that are our dependent variables, are crucial of impact assessment of contract farming. So from this table, we can see that contract farming adopters are gaining higher as compared to non-adopters by approximate 9,000 per acre. They are also producing higher yield by around 1.42 quintals per acre. Contract farming adopters have almost double land holding area. We can also notice contract farming require more inputs quantity of fertilizer, pesticides, irrigation, and also of labor. But seeds quantity used by adopters is lesser than the non-adopters because contract farming adopters use high quality seeds that require less in the quantity. Additionally, adopters have better access to credit and extension services and working on better quality seeds than non-adopters. The kernel density graph on the right-hand side also proving that the adopters are gaining higher profit and producing more wheat yield as compared to the non-adopters. This is the main result of endogenous switching regression method for the gross margin and for the gross margin. In this table, the significance of log likelihood test for the independence of equation is given in the last row of table, showing that there is joint dependence between the selection equation and outcome equation for the contract and non-contract farmers. So we can reject the null hypothesis of no correlation between the adoption and gross margin. It means contract farming participation is positively correlated with wheat gross margin. The probit regression result of contract farming selection equation represents the determinants of adoption behavior of farmers that is given in the column second and third of this table. So from this table, the result indicating that farmers with higher education, higher non-farm income, more land holding area, easy access to credit and extension services are more likely to adopt contract farming. Only distance to contracting firm have negative impact on adoption decisions of farmers, because if contracting firm is established at a long distance, then farmers will ready to sell their produce at the nearby local market. Moreover, the results of determinants of gross margin for contract farming adopters and non-adopters
Sorry to
are
interrupt, your
given.
time is over. Can you conclude?
7 minutes. This is the, so just give me 5 minutes more, please.
Yes. Okay.
This is the result for yield, but there is the same explanation of the result, but in the context of yield. This is the main result of my study. This table demonstrates that the contract farming has significant and positive impact on gross margin and yield of wheat crop. From this table, we can see that ATT implying that adopters of wheat contract farming would have reduced their income by approximately 28% if they had not adopted contract farming. Similarly, ATU treatment shows that non-adopters would increase their gross margin by approximately 32% if they adopted. Same result also represented by the yield treatment. The findings indicate that decision to adopt contract farming technology under wheat farming seems to be rational. This result shows the impact of contract farming on gross margin and yield across farmers group. So from this table, we can also clearly see that the large scale farmers are most benefited category by adopting the contract farming, that are followed by the medium and then smallholders among the adopters. The conclusion and policy suggestions. The overall result conclude that contract farming adoption is a better strategy to enhance rural welfare. It provides better alternative market to farmers and encourages them to branch out from traditional crop by providing them qualitative inputs of improved varieties. However, the returns to contract farming are heterogeneous, where smallholders could not reap the benefits to the like the large-scale farmers do. In fact, in the study region, we found from the group discussion with the farmers that contracting firm exclude the marginal and small farmers because of the scale economics. Hence, the policy recommendation of this study suggests that government should promote contract farming among the farmers, but at the same time, the inherent heterogeneity in returns from contract farming should be addressed. Smallholder institutional and structural hurdles to adopting contract farming should be removed from both the demand and supply sides. The contractive firm must bring the smallholder into the ambit of the contract. There are some references I have gone through. I would like to thank my supervisors, Dr. Kirti Ranjan Palta Singh and Dr. Pavitra Kumar Jina, Assistant Professor in School of Economics. And I also would like to thank Agri Vision 2022 and Ravenshaw University for giving me the platform to share my research. And thank you.
Yeah, so thank you, Saroj, for your presentation. So I have one question.
Yes, sir.
So is contract farming help to solve the food scarcity of our country?
Sorry, sir. What you ask it, sir?
My question is contract farming solve the food scarcity of our country?
With some extent, it can solve the food scarcity because if contracting firm do the contract farming in a systematic way, then it can solve the problem of food scarcity.
Okay, thank you.
Okay, sir.
Okay, yeah, thank you.
Okay, sir, thank you.
Okay, so next, Harish Chandar. Payal ochegi? Payal Kapoor. No.
So good evening to one and all present here. I'm Harish Chandar, pursuing my MSc Genetics of Plant Breeding in Centurion University of Technology and Management, Parlake Mundi campus. So today I'm going to talk about the topic of genome-wide allele mining to identify the genes corresponding to or conferring to folate content in rice. So domestication of rice has taken many years ago. So domestication has been mainly done to improve the production or productivity or to improve the disease resistance and other characters in the main aspects. But when we talk about nutritional quality or biofortification of rice, it has been not majorly discussed or major work has been carried out. So my topic is my topic is mainly on allele mining to identify the genes which are conferring to folate content in rice. So rice belongs to the family of Poaceae. So it is one of the major world's serves as a stable food diet for about half of the world's population, holding key to food security and playing an important role in national economy. So food with higher nutritional value is an important major importance for human health. According to dietary guidelines of Americans 2015-20, human beings require approximately 2,000 to 3,000 calories per day, among which rice provides at least 76% of total calories intake of people in Southeast Asia. Although we say rice is a major staple food crop providing 80% of the daily caloric intake of over 3 billion people, it is a poor source of essential micronutrients including folate. So this folate plays a major role in C1 metabolism and including DNA mechanism and many other functions, and it is majorly responsible for neural tube defects in humans. According to the folate is a water soluble vitamin that can only be synthesized by plants and microorganisms. The recommended daily allowance of folate, the RDA of folate is 400 micrograms for men, and pregnant women it is around 600 micrograms, and for pregnant women it is 400 micrograms, and childbearing women mostly requires about 600 micrograms according to Blanquet et al. In 2015 studies. Factors like cooking quality, storage, transportation play a major role in folate synthesis. And storage conditions also determine the folate available for nutrition. An average of 23% loss in folate content was estimated after 1 year of storage. And 100 grams of cooked rice served only 26.3 micrograms of folate, which is only 25% of the recommended daily allowance. Biofortification of stable crops like wheat, rice will provide significant scope to reduce and solve the problem of nutrition deficiency. So the main objective of this study was to develop an elite rice variety with elevated folate content and bioavailability of micronutrients to resolve malnutrition through molecular breeding methods. So what is the reason of interest? Main reason of interest behind this study is, so folate is, as we discussed before, folate is an essential vitamin. So deficiency of folate leads to neural tube birth defects, anemia, cancer, cardiovascular diseases in humans. In this direction, rice being a staple food can be a dietary source with folic acid if such a genotype is available in the market commercially. India and Asian countries consume rice, and intake of folate-rich rice in the diet would eradicate malnutrition. Thus, the present study is an attempt towards enriching the rice with elevated folate through molecular breeding approach. Background of the work, dietary folate deficiency occurs mainly due to major of the plants that lose folate upon processing and cooking. In addition to that, poor storage facilities in resource-limited countries causes folate reduction. Therefore, folate biofortification and its stabilization is essential to retain the folate content during post-harvest durations. A study by Blanquet et al. 2015 reported that the stability of the folate by overexpressing the folate binding particles protein and folate polyglutamylation by polyfolate glutamate synthetase. The result exhibited that accumulation of high folate up to 150-fold compared to wild type of rice varieties. So what is the reason of interest behind this research is deficiency of folates. Similarly, Akhtar et al.'s 2010 studies revealed that the reduced level of GGH expression, the GGH is the gene which is responsible for the folate content in rice. So reduced level of GGH expression in tomato and Arabidopsis with 99% increased tail length and enhanced the folate content by 30%. Thus, downregulating the folate destabilizing gene gamma glutamyl hydrolase in rice will lead to increase in folate due to increase in the tail length. However, most of these studies were reported upon transgenic approaches in crops. Further, the current study involves breeding program to explore the presence of novel GGH specific alleles in rice mutant population. So this is the structure of the folate molecule, and the glutamate tail which is indicated there is responsible for the folate content in the rice. The glutamate tail is reduced by the enzyme called polyglutamate, and the tetrahydrofolate which is available at the final stage is the main reason for the availability of folate in the rice. Allele mining for crop improvement. As we know now, allele mining is gaining major importance for developing a particular trait or analyzing for a particular trait in crop improvement. So allele mining is an appropriate approach technique to dissect naturally occurring allelic variations of candidate genes specific of trait interest. Brief details on allele mining. So for allele mining, we have to select first a particular trait for which we are going to work upon, and identification of rice accessions associated with desired phenotypic trait, selection of genes underlying the chosen trait, and primer designing, PCR amplifications, and we further proceed to sequence-based mining and sequencing and identification of variants and comparison of sequence data and identification of the alleles. So what is the workflow which we carried out during allele mining approach? Is we generated a mutant population in rice with BPT 5204 through chemical mutagenesis. So BPT 5204 is a Samba Masuri, which is commercially available in many commercial markets. It is a high yielding variety of long duration crop, majorly of 150 days duration and a semi-dwarf variety. And we induced chemical mutagenesis with EMS. Why we choose EMS? When we compare to other chemicals or other gamma radiations, EMS, many studies reported that EMS gives highest frequency of mutation when compared to other studies by transmitting GC to AT or vice versa, like AT to GC transmissions. And primer designing and synthesis of significant candidate gene, gamma glutamyl hydrolase, related to folate specific destability. And DNA extraction from the mutant population and PCR amplification to produce multiple copies of specific gene was carried out. And NGS, next generation sequencing analysis, to identify the functional SNPs in target genes, regions, and mutation discovery was carried out. The result and discussion, when we estimated the EMS lethal dose determination of EMS, we found that approximately 0.75% to 1% resulted in a proper lethal dose determination, LD50 value, where 50% of the seedlings were dead. The procedure of mutagenic treatment takes three things into account: the plant species, dose of mutation, and the duration of treatment. In seed propagated species, generally seeds are treated, and LD50 value refers to the mutation that kills 50% of the treated individuals. This is the graphical representation. During this analysis, we came to know that as the EMS concentration increased, the survival rate of the seedlings also decreased, and the root length, shoot length constantly decreased when the concentration of the EMS was increased. The data collected were on germination percentage, plant height, number of plants, number of plants, yield and its percentage were analyzed, and significant interaction among species. Mutagens and level of mutagens were observed for all trait under study. Optimal mutagenic dose were calculated based on survival rate and seed viability, which were considered useful in developing rice mutagens population for forward and reverse genetic studies. We raised M1 generation from uniform seeds. We treated with EMS of 0.75% and 1% EMS of genotype BPT 5204 by pre-soaking them for 12 hours in distilled water and treated with EMS and placed between folds of blotting paper to remove the excess water. These seeds were sown in a nursery beds, and the seedlings were transplanted after 25 days, and they are harvested as individual families. And from individual families of M1 generation, the M2 generation was raised, and 5 plants for each line was marked, and tissue collection has been carried out for further amplifications of DNA extraction, PCR amplification works were carried out. So M1 generation seeds of BPT 5204 were selected and sown in rice beds. After 25 days of the seedling transplanted in a well fertile and puddled soil, Suitable agronomic practices were carried out to maintain a healthy crop. 5 plants per family were randomly selected and labeled. Leaf samples were collected from the individual selected plants in Ziploc covers. Leaf sample was stored in -20 degrees for long-term storage.
Sorry to interrupt, your 10 minutes is already completed.
Just 2 minutes, sir, last 2 slides. Based upon the PCR amplifications on gel electrophoresis, we were able to detect a small amount of SNP changes in line 4 to L4 to L10. To confirm again with this, we went for the sequencing of this particular genome, and during sequencing report, we got positive results in L4, L6, and L10 genotypes. So the result indicated that L4 to L10 showed visible variations compared to other lines of mutants used. Further, the variation was confirmed with sequencing data for sequencing level of base pair changes. So sequence confirming the variation among the mutants. So we compared the sequence with one control and remaining of all mutant population. The sequence data showed line 3, line 6 and line 9, 10 showed SNP level of variations in comparison to other lines of mutant use. The results basically showed presence of SNPs and other bioinformatic analysis are underway to confirm the same and continue towards improvement of the rice genotypes. The conclusion of this experiment is mutation imposed on rice genotype BPT5204 showed morphological and agronomical differences with a variety yield. Molecular analysis showed variations among the mutant lines. The variation among lines was confirmed at sequence level, indicating the presence of novel alleles. The study would further continue to analyze the bioinformatic part of the work. The major conclusion of this experiment is most of the pregnant women are given folate as a folic acid supplement in the form of tablets. So these tablets are not affordable by middle class people or lower poverty class people. So development of a rice variety with elevated folate content will reduce the malnutrition content and will improve the nutrition qualities and status of India. First and foremost, I would like to express my gratitude to Dr. Satish Kumar, Professor and Head, Department of Biotechnology, Bharathiar University, Coimbatore, for his able guidance and support in completing my project. I would also like to extend my gratitude Mr. S. Rajendran, Managing Director, Tech City Biosciences Private Limited, Coimbatore, for providing me with guidance and facilities that was used. Also, I would like to apologize my sincere thanks to Dr. G. Pushpalata ma'am, Professor and Head, Department of Biotechnology, Centurion University of Technology and Management, for her immense support, without which I could not have succeeded by completing this project. Thank you, one and all.
In M2, whatever the plants you have developed, whether it is homozygous condition?
Ma'am, actually we have to transfer from M2 to one more generation to check whether the genes are...
No. You might have seen that it is getting segregated.
Yes, ma'am.
Have you seen? Have you observed?
Segregation of alleles are in homozygous condition in M2.
If it is segregated, then how you are picking out the plants from that population?
I can't remember.
Suppose in M1, one seed, okay, then it is segregated. Then it will be segregating. That's which plant you are selecting?
Yes, ma'am. Individual harvested plants.
Individual?
Yes, ma'am.
Okay, thank you.
Okay, thank you.
Okay, sir.
It is Saktisri Jena.
Hello, is it audible? Is it audible?
Yeah, your voice is audible.
Okay.
Your screen is not visible.
Is it visible now?
Yeah, visible.
My study is related to the physiological molecular characterization of sugarcane varieties response to nitrogen deficiency. It is done in ICR Sugarcane Breeding Institute, Coimbatore, and first of all, I will introduce myself. Myself, Satyasri Jana. I am working as assistant professor in Department of Biotechnology, Karpagavinaka College of Engineering Technology, Chengalpet, Tamil Nadu. So, the screen is changing, right? Hello, screen is changing. Hello.
Yes, it is changing. Can you please start?
Yeah. Yes. So the study is related to the because sugarcane is a major cash crop. If you see in India and all over the world, that is the second number is in India, first is Brazil. So what we actually trying to do, that is the initial study, we try to find out a type of sugarcane variety which will be which can easily grow in nitrogen deficient situations also, because what we have found out our initial studies is 80% of the applied nitrogen fertilizer is unavailable for the plant uptake. So that is why the way it has to be expected to grow, it generally doesn't grow. So we are trying to find out a variety which can grow if the nitrogen is also provided. If it is less nitrogen also, it can properly grow. So I will start with the status of the nitrogen. N nutrition actually positively affect the crop yield. Okay, so what are the areas? Because if you see the first one, the physiological and molecular characterization we have done. Mostly we have gone with the physical characterization. And then finally it will lead to the bioinformatics part. So N nutrition positively affect the crop yield with significant influence in above ground processes like leaf area, because nitrogen affects the leaf area, dry matter accumulation, photosynthesis, metabolites, which finally affects the production, which drastically reduces the production with the current sugarcane varieties. So proper amount of N fertilizer remarkably increases the tillering and the resulting in high yield. Yes, and insufficient N supply result in poor growth, narrow leaves, and reduced overall production. So if we see in this picture, there is plus N. So here nitrogen is given properly, and here it is given less nitrogen. It is written without, but very less amount of nitrogen or without nitrogen. So it will be something like look like this, which is not grown properly, and some yellow things it is visible. The sugarcane as a cash crop, if you see as a statistic, sugarcane is a value for its biofuel and sugar content. Not only we get sugar, but it is also nowadays utilized. The sugarcane baggages are also utilized for as a lignocellulosic material, and also in Brazil and various countries, they are replacing slowly petrol with a blended amount of biofuel like ethanol. So 70%, 50%, like that, slowly they are increasing and blending it with petrol. So that is why it is called a major cash crop in various countries, including India. So sugarcane in India in 2018 data, if you see, the area is 4.6 hectares, and production is 289 metric tons, and productivity is 62.8 tons per hectare. So India is the second largest producer of sugarcane after Brazil, and these are the major states. So I will quickly go to the studies, and these are the previous works done related to it. Sugarcane genotype of a mapping of populations were examined for biomass production for physiological variable under low and high nitrogen supply control. It is a Robinson et al. 2009 study, and this is also 2018 study. Physiological and molecular response of sugarcane genotype subjected to hypoxia. But our study is basically on to finding out actually what are the genes are responsible for it. That is our aspect. So objectives, I will go with the objectives quickly. So physiological evaluation of sugarcane varieties subjected to nitrogen deficiency. So we will be focusing on nitrogen because that is the major element we are dealing with. So if nitrogen deficiency, what are the varieties? They can able to grow properly in a physiological manner initially. Physiological means the leaf size will be proper, area should be proper, root diameter will be proper. So certain parameters we have taken. So expression analysis of candidate genes regulating nitrogen use efficiency in the sugarcane varieties, and identification of traits and mechanisms governing nitrogen use efficiency in sugarcane. So what are the traits and what are the mechanisms also that we are going to focus on. Then materials and methods, a simple material we have taken. These are the 5 varieties we have taken, 1, 2, 3, 4, 5. These all are good varieties, but among them we are trying to find out these all 5 varieties. Actually, it is currently supplied to the various farmers. They are utilizing it, but still, what are the varieties we are trying to? What are the varieties among these 5 best varieties are can perform in a nitrogen deficient condition also, that we really want to find out. So 5 varieties are taken. So our treatment method will be the controlled flooded with nutrient solution. So we have given nitrogen in ample amount and in control, and deprivation of nitrogen 0%. We have not given any nitrogen to other parts. So if we see the methodology, if you go with the methodology, because these are the varieties and this is the control. Control has been given with nitrogen and deprivation of nitrogen, and those who are under study, the same plants, we have not given nitrogen. So plant growth analysis, if you see leaf area meter, we have checked, and root scanner also through, we have checked the how much actually, how the diameter of the roots and all that also we have checked, and thermal imaging camera. So for the chlorophyll amount, to find out the chlorophyll amount, SPAD for the finding out the anthocyanin amount also we have done. So chlorophyll content. So these are the biochemical assay for the metabolite concentration. So this is for the physical aspect, and these are the tests we have done for the biochemical assay, like finding out the extraction of sugars, non-reducing sugars, reducing sugars, total sugars, how much is. So various methods we have followed, and starch content, total soluble proteins, total. So like that, a lot of parameters for different aspects we have taken, like antioxidant capacity and plant water status. For that also related water content, membrane stability index, lipid peroxidation, cattle age, peroxidase, because these are the major enzymes for the growth and stability of the particular organism. So now if you see that RNA isolation and gel electrophoresis also we have done to find out actually for various parts, like we have taken RNA from the leaf, root, by various parts to find out actually to see the stability of the RNA, because that will ultimately decide the further processing of like translation to a particular protein and further processing and further survival of the plant. So that also we have done by gel electrophoresis to find it out whether it is stable or not, with comparison with a controlled one. So suppose we have not given nitrogen and we have taken the leaf, and we try to find out that whether the RNA is stable or not, that particular variety. So these are the results I will discuss. So photosynthetic pigments and efficiency, if you see here, influence of nitrogen deficiency in SPAD chlorophyll. So these are the 5 varieties I talk about. These are the 5 variety, and yellow represents the control one, and the green represents the nitrogen deficiency one. So in this case, we are checking the chlorophyll photosynthetic pigment contents. So here if you see that CO 10026 and CO 86032, they are somewhat showing significantly without nitrogen also the content is better compared to others. So but with nitrogen it will be definitely best, but we are dealing with the where nitrogen concentration is less, still they are able to perform, okay, in the biomass and various physiological way. So influence of nitrogen deficiency in chlorophyll A. So here if you see the CO 10026 variety, it has shown some tremendous values, like it is more than 0.2. So this is the chlorophyll concentration in mg per grams. So these are the things. Then influence of nitrogen deficiency in chlorophyll B. Here also if you see that CO 10026, it is also coming. It is showing the highest values in case of pigment and efficiency. And next, if we go to the next slide. So in this case, the influence of nitrogen deficiency in canopy meter. So canopy, if you see, so in this case, it is almost with nitrogen deficiency also, if you see, it is the canopy temperature degree centigrade, and sugarcane varieties if you see, then CO 10026, it is showing some good values, and this one is also showing CO 86032. So similarly, chlorophyll fluorescence, if you see, then in this case also chlorophyll fluorescence, in this case also CO10026. So like that, we have done many parameters we have taken from various aspects, so that we should be finally reach to a conclusion that it should which one is the best one and which can be further taken into consideration for bioinformatics studies and finding the respective gene for that particular. So we can go for a transgenic plant, and also we can go for breeding also, so that we can finally get that type of plant which can withstand all these type of parameters. So influence of nitrogen deficiency in total carotenoid also, here also CO10026 also has a tremendous value. It is among all, if you see, it has the highest value. So though they are lower than the obviously if you are supplying nitrogen, it will be better, but we are talking about here if nitrogen is deficient, okay, then which can survive and which can give us better biomass in a physiological way, because ultimately the biomass and the sugar content, all those things will decide how good is our product here is our cash crop, sugarcane. So here if you see also same CO10026 also in case of anthocyanin, if we take there also it will be a better value. So like that also we have lipid peroxidation, superoxide dismutase and peroxidase activity, catalase activity. If you see everywhere CO106, and here also it has shown a very good value. If you have seen here CO86032, it has also shown very good value, and here also if you see CO86032 also shown very good value in lipid peroxidation and peroxidase activities. If you see this one is better, CO86032, this one is better. So overall, if we see all the 5 varieties which are currently in use, then CO10026 and this one CO86032, CO86032 and CO10026, these two are showing some tremendous characteristics in nitrogen deficient condition as well. So similarly, if you see the metabolites contains also, so in this case also same CO6002 and CO10026. In case of the protein in the sample, also it is coming, these two are coming somewhat near to each other and highest values compared to the others. So finally, so similarly, the performance of the amino acids under deficiency and performance of the carbohydrate content under deficiency, performance of the reducing sugar under deficiency, N deficiency, and performance of the non-reducing sugar under N deficiency, nitrogen deficiency, and performance of the starch content under nitrogen deficiency. So similarly, we have studied all the aspects. So the plant morphology, so those are physiology, and we have seen some metabolic conditions also. So now if you see the morphology as a whole, then change in the plant height of the sugarcane varieties at deficiency. So plant height also if you see, then here it is getting less affected in CO10026. Similarly, if you see
Sorry to interrupt you.
hello
Time is over.
yes ma'am, just I will conclude it.
Yes, please.
So a lot of studies we have did related to the morphological and physiological aspects, and finally we have done the RNA integrity, because that is also most important. RNA integrity means whether it is getting degraded or not. So if you see in this case, so finally we got that these two varieties are actually more important. And if you see here, second number, fourth number, and the sixth number and eighth number, there we are actually getting that they are not getting degraded. So somewhere we can take this variety as a our consideration among all the 5 varieties. And conclusion, we can say that from the physiological and morphological characterization of nitrogen deficit varieties, the crops CO86032 and CO10026 performing efficiently compared to the other varieties. So we got that one. And 2 varieties, these 2 are identified as promising under nitrogen deficiency, and gene expression of the respective crops we need to study. That will be the future work related to bioinformatics. We have to go for the next generation sequencing to find out the genomic sequence of, which is very complex in case of sugarcane. That will be the further study. Thank you.
The presentation was good, appreciable. Please let me know what is the chlorophyll fluorescence at maximum level. You have given FV, FM, I am asking. No, you are going back.
Just a second.
Not here, next slide. Yeah. FE by FM you have given, what was the maximum? Previous slide. No, not here. Previous. One more. Here. I can see that there is chlorophyll
Yes, yes.
by FM. What was the maximum, if you remember?
Around 0.9, madam.
Have you analyzed non-quenching and quenching?
No, no, madam. Actually, there was facilities like we are getting the samples and all. There are actually authenticated people. They do it. We generally get the data.
So how did you analyze this chlorophyll fluorescence then? Along
We
with
do the. There is a procedure. We go with it.
Riker is there.
Yes, madam.
If you take up this Fe by FM, obviously this quenching and non-quenching also can be recorded.
Yes, madam. But we didn't do that.
Okay. Anybody is there who can ask questions to him? Any questions?
Then you were telling that you have applied nitrogen.
Yes,
What
ma'am.
is the dose of the nitrogen you have applied? What is the dose of the nitrogen?
Nitrogen dose.
Huh?
You are asking about nitrogen dose.
Yeah.
We went with actually 3 to 4%. Basically, for the plants we have studied, we actually didn't apply nitrogen.
Actually, commonly used dose of nitrogen, how much, you know?
I know, madam. But we didn't go with nitrogen, or we have applied very less amount for the.
Less amount means what is the dose? Less amount, science, less amount. It is not actually advisable to say.
We went with the recommended NPK values.
Then you are telling that you have only applied the nitrogen.
Only nitrogen.
Then what is the dose? Like how in rice, like 30, 40, 30, 30, like this 40 is suppose nitrogen, in which form also you have given?
We have actually hydroponics only. We went with hydroponics. This is soilless culture.
But yeah.
Yes, madam. We went with hydroponics starting
No,
only.
but you are showing this photograph.
Yes,
It
madam.
contains the soil.
No, this contains soil, but this is not actually soil. This is basically like something like you can say sand type of things.
Sand
So
like, okay, but that should
we
be
have to plant it somewhere because sugarcane we cannot just do like hydroponics and put. We have given some.
What amount of nitrogen you are applying? You should tell. With nitrogen, without nitrogen.
Yes, ma'am.
Little amount, nothing. Science cannot tell that little amount like. Okay. If you don't know, okay, stop it here. Thank you.
Yeah, so I have only one question. So how you can calculate it that carotenoid contains? Actually, madam asking that what is the quantification of nitrogen you are taking in your experiment?
Yes, ma'am.
What is the
Yes,
exact
sir.
value?
Yes, sir.
I mean, asking actually the value, because science without value is none. Because how you can calculate how you can say that this much of data was
Because after experiment we got these values.
Okay, sir. Then can you tell me, sir, how you can calculate that your carotenoids?
Standard graph. We got the standard graph and we got it from there.
Yeah. Thank you, sir. Thank you. Your presentation is good, sir. Thank you.
Thank you.
So next, is your Rageshri Swai. Are you there? She is from Revensa. Pragya Raut. Both are Revensa students. So shall we stop, ma'am? Okay, so thank you so much. So we will close this session. Thank you all.